right, good morning, fam. Good morning, good morning, good morning. All right, so today is devoted to repro. We'll also, in lab, we'll review some reproductive anatomy and we will do some case studies. Gonna be a busy day. Still like to get done, I don't know, a little bit early if I can, just to give you guys a little bit of reprieve. So let's get rolling. Hopefully, I only got a few people that ended up texting me their flow charts for the reproductive systems, and every single person had made mistakes. And I was able to, I hope, get everybody that did send me pictures of their flow charts for the repro anatomy. I was hopefully I was able to kind of set you on the right path, and so you were able to either fix them or whatnot. We will look at that flowchart today. We'll do something like that. Kind of one of the first things that is important for you guys to understand, of course, with the reproductive system, because structure determines function. So I hope you guys were able to study at least a little bit of that reproductive system like I'd asked you to. So cool. Let's see, reproductive anatomy. So what makes reproductive anatomy and physiology different than the rest of the systems? I like it. Yeah, not necessary. It actually is about homeostasis, though, Melissa. There's a lot of homeostasis in the reproductive system. A ton. Nope, not an oops. It's okay. I see what you mean. Because you're talking about not about homeostasis that regulates life, like whether or not you're living or not. I understood what you meant. I just wanted to clarify, right? It is about homeostasis because it still requires so much homeostasis to run your reproductive system. But it's not about survival, right? It's only about reproducing your genetic information, passing that on to your offspring. That's what the reproductive system is all about. I don't know why this is opening it up over here. Come on, meow. So when we're dealing with the reproductive system, we're dealing with actually genetics, which is a little off-putting for some people. Because we're actually talking about passing on genetic information. And therefore, what we're talking about are specific types of cells that are all about passing on genetic information. Not every single cell in the body does this in the same fashion. There are two types of cellular division. You guys may or may not remember from a long time ago, somewhere in high school, promptly forgot it, maybe took it in a college class, promptly forgot it. And that's mitosis and meiosis. Do you guys remember those two words? I mean, at the very least, are they in your brain somewhere and you're like, yep, they make no sense to me, probably. I constantly get that where students are like, this is definitely something that I've learned and I just don't know why it's relevant. I don't really care about it. It'll make maybe a little bit more sense after the beginning of today's lecture. We'll definitely refer more to mitosis and meiosis, and you're going to get reminded on why it's relevant and how it's important to physiology. But ultimately, most of the cells of your body are dividing and making exact duplicates of themselves. This is called mitosis. That's 99.9% of the cells of your body. That's trillions upon trillions of cells that are going through mitosis. And mitosis is about creating these carbon copies, these exact duplicates, these mirror images. The other cells of the body, the other 0.1% of the cells of your body, are going through meiosis. Meiosis produces sex cells. And that's what we're going to be dealing with here with the reproductive system. Production of sex cells. And sex cells are called gametes. Gamete, gamete. So long story long, when we're talking about what is the function of the reproductive system, a lot of people will say to reproduce, but what does that mean? It means that we produce gametes. The one major function of the reproductive system is to make gametes. Everybody write that down? The male gamete is the spermatozoa. The female gamete is the oocyte. I love that word. It's literally two O's, and it's pronounced oocyte, not oocyte, because that'd be weird. Not that oocyte isn't a little weird, but it's more fun to say. Everybody, what is the primary function of the reproductive system? What you're going to learn is that the production of gametes requires both mitosis and meiosis for its function. And we're going to have to go through gametogenesis. We're going to have to go through the creation of new gametes in both the males and the females. It's going to be a big part of what you guys learn. Is that clear? Awesome. Another thing you guys are going to be learning about for the physiology of the reproductive system. Man, there is so much going on from, especially in the female reproductive system. Male reproductive system is pretty benign, ultimately, when it comes down to it. I mean, make sperm, have an erection, and ejaculate. I mean, it's a really silly system when it comes down to it. Female reproductive system is super complex. You guys probably already knew this. Everything from the oogenesis, where you're going to get these primordial oocytes that are produced before you're even born. And they're going to be frozen in stasis. And they're going to have to come out of stasis at different times and go through this crazy developmental stage, only to get ovulated into void, into space, into the peritoneum. It's like literally shooting something off into outer space and saying, all right, good luck. You can figure this out on your own. It's bizarre. And then that oocyte is going to end up getting transported to the mothership, which is the fallopian tube. It's going to take a bit of work to get it up in there. There's so much going on just at the beginning, let alone during pregnancy. We are not going to cover pregnancy. Not enough time, and it's not part of the Common Core objectives. It's not something that your medical and nursing programs want you to learn before getting there. So it's not part of our objectives in this class. We will get you to fertilization, and that will be as far as we go. Okay, we'll talk briefly about implantation, but we're not going to talk about fetal development. That's an entire class in and of itself. It is super complex. Sound good? So what are we going to learn? We're going to learn about gametogenesis, and part of that is getting it to the point of fertilization. Awesome. The last thing we're going to focus on is for gametogenesis, this is going to require a lot of hormonal control. We're going to be talking about the homeostasis. Yeah, I'll show you no fun book. We're going to be talking about how the endocrine system helps control the homeostasis of gametogenesis. If we have too many spermatozoa, what do we know the body's going to try to do? Increase, decrease, no change spermatozoa production. Pretty simple. It's pretty simple. Okay? Female system's a little bit more complex, but we'll deal with that. There's a lot of big, huge cycles that have to go on. We're going to cover two more things. One, because they're part of the Common Core objectives, but two, because they're also really cool and really relevant. One of them is called sexual differentiation. Sexual differentiation. So different is a key part of that word, differentiation. This is the only part of fetal development that we are going to cover, sexual differentiation. Right around 6 weeks of fetal development, this is when sex organs start to develop. We're all born with the exact same sex organs. Every single one of us looks like a tiny little frog with the exact same sexual organs. It's called a bipotential system. Everything is bipotential, which means it has two potential outcomes. Depending on the hormones that are in the system, it could either become male or it could become female. And all it changes is all it takes, I guess I should say, is a slight deviation in the hormones that are present for the outcome of sexual differentiation. Trying to emphasize the point that just because you are XX doesn't mean you're going to be female phenotypically. Just because you're XY doesn't mean you're going to be male phenotypically. It's really, physiology is so complex that just a few small changes is all it takes. Yeah, not YY. No such thing as YY. Thank goodness. Yeah, YY would be a bizarre one. The Y chromosome, I always make this joke. I typically would make it later, but why? Do we have a Y chromosome? Why? Literally, the Y chromosome is one of the most dumb chromosomes on the face of the planet because it only holds just a couple of genes, and one of the genes it holds is called a SRY, which means the sexual differentiation protein. Basically, is going to be produced from it. Without the SRY, there's no male reproductive organs. But the X chromosome is what contains everything necessary for life. The Y chromosome is just to make a penis. It's pretty funny. So let's see. So everybody, we got a list of 3 things we're going to be learning about so far. Gametogenesis, and that's going to include, is it 3 things already, I guess? So I guess we'll separate that. So gametogenesis, we'll literally just talk about how spermatogenesis and oogenesis is created. Then we're going to talk about hormones that help control gametogenesis. And then we're going to be talking about sexual differentiation. And the last thing that we're going to refer to, but be tested very little on, but it's the end of the semester, is sexual arousal, because it is a Common Core objective, and it's kind of funny to end on that. I can't start a semester without emphasizing everything about the autonomic nervous system and not end the semester talking about the autonomic nervous system. What do you guys think controls sexual arousal? Is it voluntary or involuntary? Sexual arousal is a very involuntary thing. Therefore, what part of our body's amazing systems controls it? Parasympathetic nervous system for the male and sympathetic nervous system for the male, and only the sympathetic nervous system for the female. So we'll talk about that. Males and females are not the same, the way that they are sexually aroused. We already knew that, but there's actually a physiological reason for it. All right, everybody clear on what their study list is going to be over the next couple of days? You don't have a lot of time to study this material, so I wanted to get that study list ready for you right now, so you're not frantically searching for it later. And of course, one more thing on the list, of course, flowchart of these systems. So if I ask you to go through that flowchart for spermatogenesis, you're going to start where spermatogenesis occurs, and then the question will say, you know, follow this flowchart all the way to ejaculation. So now you got to go through a flowchart all the way from those seminiferous tubules all the way to the external urethral orifice. The people that sent me their flowcharts already worked on that. So if you've already worked on that, you got a big part of your studying out of the way already. If you haven't done those flowcharts yet, this is when I pull out that GIF, you know, the shame, shame, shame GIF. Shame, shame, shame. You need to be working on these things, pretty please. Not a lot of time left. And then for females, it's going to be from the process of oogenesis until you can either think about it as childbirth or until the menstrual cycle. Either way works for me, but you're basically going to be going all the way from the ovaries to the external vaginal orifice. So these flowcharts aren't too complicated to get through. All right, is everybody clear on what they need to know? And then we'll get started on it, and we're gonna fly like a bird. Make me a bird so I could fly far, far away from here. Awesome. Good to hear. Okay, let me get my pad out. The reproductive system is very, quote unquote, simple in terms of what is primary and what is accessory. The primary function of the reproductive system is to produce gametes. Therefore, only one organ does that job, and it's called the gonad. In males, the gonad is the testes, and in females, the gonad is the ovaries. Everything else is accessory. Again, you know, great joke. Yes, the penis is just an accessory. So you guys, you know, bring that up in a Thanksgiving conversation and be a great one. Were you guys laughing about females? It means that, you know, even the uterus is just an accessory. But yeah, it's a pretty funny one for sure. Yes, most people on the street, what's the primary sex organ for the male? They're going to definitely say penis. So what does that tell us about the male testes? This is where gametogenesis occurs. True story? And in the females, that gametogenesis will occur in the ovaries. So let's do a flow chart for the male reproductive system. I'm not going to focus on the same stuff that's already in this lecture, right? Going through this, you guys should have done this already, but you'll notice it's a flow chart, right? Where I talk about this male reproductive system all the way from, well, what's going on out here in the scrotum? You know, and it's always been a curiosity until I took a reproductive physiology class. Like, why are the male testes housed outside of the body? I mean, it makes no sense at all. There are definitely some animals where the testes are inside their bodies, but all mammals, the testes are outside of the body. So what's going on there? Didn't seem very efficient to me. Well, the reality is spermatozoa are extremely sensitive to temperature when they are being created. So immature spermatozoa, they don't have a hard helmet on yet. They're not ready for World War II yet. Yeah, so someone can kick you. Right? They're just this infantry person that's going into boot camp. They don't even know what the heck is going on yet. And what the whole function of the rest of the organs down here inside of the testes, inside of the scrotum do, it's to make those spermatozoa mature. So as it's going through this system, it's going to make them mature. So now these mature spermatozoa, where are they going? They're going up into the body cavity. Why is that? Again, that's because these immature spermatozoa are extremely sensitive to temperature. If they make it to body temperature, they will die. It's crazy. If they get too cold, they will die. So we have all sorts of things to help with temperature control. And this entire slide is over the temperature control of the scrotum. There are two major muscles, dartos and cremaster muscles, that are down here. Both of these are smooth muscles. One of them runs up longitudinally up the spermatic cord. So it's going to cause the ascension and descension of the testes. That's your cremaster muscle. You know, there's a joke, of course, cremaster muscle. So if that helps you understand this muscle better, this is the master for making sure your spermatozoa don't die. This was also the name of my softball team for like 5 years in college. Nobody else thought it was funny. And it's like people didn't understand what a cremaster was. We all laughed. Yeah, I'm a big dork. What can you do? And what do you guys know? Is it going to be warmer closer to the body or further from the body? Is it going to be warmer closer or further? So as it's cold, then these testes are going to ascend up closer to the body to help warm them up. Awesome. Now when it gets too hot, then it's going to descend further from the body to help cool them down. Sweet. What about this dartos muscle? The dartos is inside of the scrotum, inside of the house for the testes. It's actually lining that integumentary system. So when this one contracts, it causes wrinkles. What do we know about wrinkles? What's going to happen to surface area as something wrinkles up? Surface area increase, decrease, no change. It's going to increase. It's going to increase. The more wrinkles something has, the more surface there is. Does that make sense? And surface area traps heat. So that's another thing. Surface area does so many things, from diffusion to absorption. But something else it does is it traps heat. And if you're thinking of like comparative anatomy, you know, animals of the north all have huge surface areas. Because the more surface area an animal has, the more wrinkled a walrus is, for instance, the more heat it can trap, even though it's also got all that blubber and everything too. So when it's cold, those dartos muscles are going to contract, cause wrinkling of the scrotum, help to hold in heat. Get it, got it, good? Of course, we know this as shrinkage. So, you know, that helps. The pampiniform venous plexus. This one is super cool. I love the pampiniform venous plexus. Let's briefly talk about it as we go into our flow chart. We have enough light. So if we're talking about the scrotum, really you guys need to understand that there are three major parts of the scrotum that help with temperature control. All of it really helps with temperature control. Scrotum. But the primary anatomical features of the scrotum that help with temperature control are going to be the cremaster, dartos, and this crazy thing called the pampiniform venous plexus. Now, you guys have seen this word plexus a few times, and it just means network. So it's a big network of veins. Pampiniform venous plexus. And it wraps around the spermatic cord. All right. So what ends up happening here is something very similar to the counter current exchange in your kidneys, where if I had blood come straight down, we got to remember that we're warm-blooded animals. Okay, warm-blooded animals means that our blood is warmer than our body temperature. The core of our body warms this blood up past 100 degrees Fahrenheit. Okay, random, but I'll check it out later. So this blood going down into the testes is going to be over 100 degrees. If that makes it down into the testes, what's it going to do to these immature spermatozoa? Yeah, it's going to kill them. We can't have that, right? So normally you just have hot blood coming down, and then that would cool down as it got out towards the extremities, and then you'd have cooler blood coming back up the other direction in a vein. This is what happens why your fingers get cold so fast if you're swimming in cold water, for instance, right? It's because you're cooling your blood down as it goes up, as it goes out towards the distal aspect of your extremities, and then it's going to be cold, bringing cold blood back towards your core, which now starts causing things like hypothermia and stuff like that. But this is not what the pampiniform does. Instead, what the pampiniform does is we got a venous return that wraps around the artery that's descending. So the artery goes down and the vein wraps around it going back up. And what do we know about everything in the universe? It's trying to go from an area of everything's trying to go from an area of high to low, and that includes heat. So heat is trying to go from high, and as it's descending down towards the testes, if it's got all of this cooler blood returning to the core, then the heat from the artery is going to dissipate, and it's going to go into the veins that are returning back to the core. So the venous blood here in the testes is a little bit warmer than it would be anywhere else, and when we make it out to the testes, that blood's going to be cool enough not to kill the spermatozoa. Pretty cool, huh? Smart. By the way, this is how dolphins and whales and other mammals survive in the seas. This is why they're warm-blooded animals and they don't get hypothermia, is because all of their blood is like this, out to their fins, out to their tails. All of their blood creates these huge venous plexuses that cover their arteries. All right, cool. Now, that way that cold water doesn't cool down their blood and return cold blood back to their core. It's really a brilliant way to do things. So you guys make sure you guys understand that temp control of the scrotum. All right. So as flow charts go, everybody work with me. Just listen. There's a lot of words on these slides. This is the testes. All of this, really. This little white egg, this is a hard-boiled egg. but instead of yolk, it's got top ramen. So I got a hard-boiled egg white with top ramen inside of it. The hard-boiled egg white is called the Tunica albuginea. Albu, as in albino. This is white. This is a white tunic. The white tunic is like proteoglycans and stuff. It's actually a hard shell in essence, made of proteins and carbohydrate kind of core. The top ramen is seminiferous tubules. There are meters upon meters upon meters of seminiferous tubules inside the male's testes. This is where spermatogenesis occurs. So if on a test I ask you, where does spermatogenesis occur? The answer is not testes, because I need you guys to be more specific than that. What's the answer? Where does spermatogenesis occur in the male? Top ramen. Yeah. Yeah. The answer is seminiferous tubules. So if I have you start a flow chart where the flow chart says, draw a flow chart from spermatogenesis to ejaculation, you're going to start in the seminiferous tubule. Is that clear, everybody? Awesome. From the seminiferous tubule, this immature at this point. This is very immature. What does immature sperm even mean? Let me just, I don't have a slide of it here. Sometimes I worry about writing these things. Yeah. So I never really know what's going to come up here. I'd like to go to images. We'll see. Let me see. Here we go. I got it. You got to be really careful. Here we go. Anatomy of sperm. There we go. So we got to remember what sperm actually is, right? And you're not going to be tested on sperm anatomy by any means. But sperm, you have the nucleus of the sperm, the acrosome up here in the front. You got mitochondria held in kind of this middle of the body, the mid piece, and then the tail is just a flagella. And this flagella, it can wriggle and move and therefore propel the spermatozoa forward. The immature spermatozoa, it doesn't have this hard helmet on, okay, this acrosome on there, and the flagella can't move yet. So it's basically just kind of a house, this limp house for the genetic information. Genetic information is held up here in the head of the spermatozoa. All right, so if I have an immature sperm, I'm going to need to make sure that that thing gets mature. I'm going to put a hard helmet on it. I'm going to make that tail start to move. Does that make sense? Well, that's the function of the rest of the testes. So as it goes through the rest of this system, it's becoming mature. Is that clear? I think it is. So spermatogenesis occurs out in the seminiferous tubules. This immature spermatozoa now is going to enter the ret testis. How does it do that? All of these tubes are going to be lined with cilia, which create a current, which now move these immature spermatozoa through this portion of the system. From the ret testis, this little maze-like structure of top ramen here, that's going to pass that immature spermatozoa up into this funny little slug. It looks like a slug. I mean, look at it like this. Look at that. Or a worm. A real weird wrapped worm. This is called the epididymis. It's a weird word. You guys need to practice writing it because of its spelling. Students misspell this thing all the time. But this epididymis is actually about 18 to 20 feet long. It's all of these coiled tubes. It's very, very long. People think spermatogenesis is like immediate because males are producing, a billion new sperm every day. I'm not exaggerating either. It's some obscene number. It's billions of new sperm every single day. But that doesn't mean that they're ready to be used the day that they are produced. In fact, it takes months. Okay, it's going to take well over 30 days just to get through the epididymis, to go from the head of the epididymis through the body down to the tail. It takes over 30 days of maturation to go through these 6 meters of epididymis. But by the time it's done, these spermatozoa are ready for war. They got their hard helmet on. Man, they've been running, they've been climbing. Therefore, their flagella is now just propelling them like crazy. They're ready to go. Why am I using war as my analogy? Do you guys think the female reproductive system is, happy to see spermatozoa? Is it like, come on in? No. Female reproductive system is a war zone. It's trying to kill every last spermatozoa that gets in there. It only wants the strongest soldier. End of story. It's literally going to try to slaughter everything that's in there. Highly acidic environment, lots of enzymes. It's crazy in there. So these spermatozoa have better have gone through the most rigorous of boot camps before they enter that female reproductive system. Cool. All right. Hopefully everybody's on board. The analogy always has worked for me, and I hope it works for you guys as well. From the epididymis, now we have a mature spermatozoa that's going to enter the spermatic cord. The spermatic cord is going to carry it up to the main portion of the body. Well, that's going to pass through the vas deferens, or the ductus deferens. If you've ever heard of the term for, when a male goes to get snipped, it's called a vasectomy. That's because ectomy means to cut. Vas is what they're cutting. They're cutting the vas deferens, so they called it a vasectomy. Make sense? All right, cool. We got a flow chart going here, don't we? So we're going seminiferous tubules, into the ret testis, into the epididymis. We got the production of immature spermatozoa that's getting mobilized and going through a maturation process through the epididymis. Finally, it's going to enter the vas deferens. Now, the vas deferens is a part of the spermatic cord. So you're going to want to have like a little bracket for spermatic cord. You're going to want to go through what is the anatomy of that spermatic cord. That's an important thing, right? So things like the pampiniform venous plexus, boom, that's here. Nerve supply, boom, that's here. Better have some nerves that go down there. Is that clear? So that's how you're going to break apart some of these flow charts, is you're going to list the important parts of what's going on. Next to seminiferous tubule, you can write spermatogenesis occurs here. Epididymis, you can say maturation occurs here. So you can take your flow chart and you can elaborate on it, and it really helps. But the spermatic cord is kind of an important structure, because that spermatic cord is going to allow for the ascension of these immature spermatozoa into the organ, into the abdominal cavity. But in order for that to happen, there's going to have to be a space, a gap, an exit point from the abdominal cavity down into the testes. This is an exit point that females don't have. We got to remember, the abdominal cavity here is filled with intestines. There's so many organs in here, and normally this point right here, right, we wouldn't want this to be open. We don't want this to be a weak point of contact whatsoever. There's lots of muscle in the pelvic floor down here that helps hold all of this together. This opening right here is called the inguinal canal. Inguinal. It's called the inguinal canal. There's not a good image for it here. We'll end up kind of doing it in our drag and drop. But this space, if I ask you what space is here, it's called the inguinal canal. I-N-G-U-I-N-A-L, the inguinal canal. And that inguinal canal is what allows for the vas deferens to enter the abdominal cavity. So let me ask you, what if that inguinal canal was weakened in any way? Now all of a sudden I have a weak point of origin that's just inferior to a whole bunch of intestines. Where are those intestines going to go? Down into the inguinal canal. What's this called? Who am I? Yeah, this isn't just any type of hernia either. This is an inguinal hernia. These guys are extremely painful because the intestines drop down into the scrotum. They're extremely painful. In fact, it caused the scrotum to swell up multiple times the size that it should be as it descends down. You had one? Dude, my brother had one too. And unfortunately, the way that, oh yeah, look at this. There's an inguinal hernia. How have you not gone to the doctor before now? I'm just saying, you know, I don't know. I don't. Some people let it go a little too far. 700 pounds leg press. Yep. You know, yeah. No comment. That's a lot of weight. I can't believe nobody went, wow, to that picture. It's insane what can happen with an inguinal hernia. Most of them aren't going to get to that point, of course. But yeah, this happens a lot in people that try to lift too many weights, which is one of the reasons why we wear those hernia belts when, you know, that's why people are required to wear them in certain jobs. Yeah, you guys were just in shock. So that inguinal canal is an important little anatomical piece. I will always test you on it somehow, whether it's in the lab practical or in the lecture portion. So keep that in mind. So now let's look at this picture and let's stop looking at what's here and start looking at the flow chart that we just wrote. Here's that tunica albuginea. So inside this tunica albuginea is the seminiferous tubules and the red testis. That immature spermatozoa is going to enter this slug, the epididymis, and it's going to become mature over a process of over a month. Finally, it's going to be able to ascend up the vas deferens, which is part of that spermatic cord, go through the inguinal canal into the abdominal cavity. Once it's inside the abdominal cavity, this is the bladder right here. This vas deferens is going to wrap around. This is the posterior aspect. We're looking at the patient from the back. So this is going to wrap and it's going to move posteriorly to the back of the bladder. And I love this picture. It's a great picture for looking at the anatomy because it's intact on the right and it's been dissected away on the left. What do you guys notice about the vas deferens? Is it turns into a pea pod. Yes, no, maybe so? Kind of looks like a pot of peas. The normal vas deferens is just an open tube. This is just a lumen. It's just a straw. But here, we now have open spaces. These are called ampulla. So this portion, no, definitely not to a sphincter yet. This portion of the vas deferens is called the ampulla of the vas deferens. Please don't just write ampulla on a lab practical or on the lecture exam. This is an ampulla of the vas deferens. And these are little chambers surrounded by smooth muscle. These little chambers, these little ampulla, are where mature spermatozoa are stored. And during an ejaculation, what's going to happen is the smooth muscle around this ampulla are going to contract, and that's going to push a small portion of mature spermatozoa into the next tube, which is called the ejaculatory duct. We'll come back to that. But then the mature spermatozoa from these ampulla, they're going to move up to the next ampulla. The spermatozoa that were here move up here, so forth, so on. So if anybody's ever wondered how a male can have multiple orgasms in one day and still be fertile, well, that's why, is because those mature spermatozoa, we're not ejaculating all of them in a single ejaculation. There are multiple sources for mature spermatozoa. Now, from the ampulla of the vas deferens, it goes down into this tiny little tube. It's almost easier to see on this side. It's right at the tip of the arrow. It's very short. It's just right, boom, there. That's it. It's very, very short. Yes, no, maybe so? Right at the tip of the arrow. This portion right here is called the ejaculatory duct. Only this portion right here is called that ejaculatory duct. And so a few people sent me flow charts, and they said that there was spermatozoa that went from the ampulla of the vas deferens, and that went into the ejaculatory duct, and then they said, and then that goes into the seminal vesicle. Well, that's not how the flow of information works. They're separate flow charts. This orange thing that you see right here is called the seminal vesicle, or the seminal gland, okay? The seminal gland. So let's take this vas deferens, let's make it into the ampulla of vas deferens. That's where these guys are going to be deferens. That's where these mature spermatozoa are going to be stored, and they're waiting for an ejaculation to cause contraction of those ampulla. Now, the next place they go once they're ejaculated is into the ejaculatory duct. Sweet. That's just spermatozoa. Semen has a lot more than just spermatozoa. In fact, less than 10% of semen is sperm. Most of semen is fluid. Fluid that helps to protect that spermatozoa. A bicarb-rich fluid. Bicarb-rich, wow, right? We're protecting the spermatozoa against acids in the female reproductive system. Make sense? The large majority of this fluid, over 60% of this fluid, comes from the seminal vesicle or seminal gland, same thing. Okay, this is what produces most of your semen. This is seminal fluid that comes out of the seminal vesicle. This is going to go during an ejaculation. It has smooth muscle too. It's going to contract and it's going to send seminal fluid into the ejaculatory duct. So it's just a simple divergence of your flow chart. Don't have it lead from here to here because that's not the direction that the arrow goes. Does that make sense? For everybody that was like, how do I separate these flow charts? Well, that's how. A flow chart does not have to be linear. You can have multiple angles in a flow chart. That ejaculatory duct is now going to enter the prostate gland. And when it enters the prostate gland, you'll actually see here that we have a tube inside of that prostate gland. This prostate gland is just inferior to the bladder. Yeah. The bladder, urinary bladder, contracts and urine goes through the same tube. This is the urethra. Everybody feel pretty comfortable with the idea that this whole tube down here is called the urethra? Yes, no, maybe so? But there are different portions to the urethra. There's a portion of the urethra that goes through the prostate. This is called the prostatic urethra. There's a portion of the urethra that goes through the urogenital membrane, the urogenital diaphragm muscle right here. So this region right here is called the membranous urethra. And then there's a portion of the urethra that goes through this darker blue area, which is an erectile body called the corpora spongiosum. You guys are going to learn that in just a bit. But that's why they call this the spongy urethra, because it goes through the spongiosum. So when you're writing out this flow chart, please be specific. Okay, we're going to have a prostatic urethra, a membranous urethra, and a spongy urethra. Please write all three into your flow chart. Deal? I hope that you guys studying beforehand is making this a little bit easier, and that I'm just helping piece together some of the puzzles that you might have had trouble with, or just redundancy, therefore you learn. Prostatic urethra. Well, this is in the prostate. The prostate does something. The prostate is actually a pretty important gland. And it's going to be secreting more of that seminal fluid. It's prostatic fluid that goes and joins the rest of the semen. So about 60% of your semen comes from the seminal vesicle. Less than 10% is spermatozoa. The rest is going to be prostatic fluid. Prostatic fluid is very similar to seminal fluid. It's also alkaline, right? It's a basic environment. It's got a little bit more energy, though. Energy for spermatozoa. You may have read this, you may not have read this, you may know this already, I don't know. But spermatozoa requires energy just like the rest of the body. Okay, it's got mitochondria just like the rest of the body does. But the primary energy source is a little bit different than the rest of the body. The rest of the body requires glucose, whereas spermatozoa, they require fructose. They're one of the few places in the body that actually utilize fructose directly for energy. So we store tons of fructose in that acrosome of the spermatozoa, right? Basically, you know, now we're going from an analogy of war to, you could also use an analogy of, like, you know, the bear helmet with the little tubes going down to the mouth. But it's full of fructose instead. So the prostatic fluid also has fructose in it to help with the motility, the movement of spermatozoa. There's something else going on here. Do you guys see these tiny little bulbs right here? They look like tiny little turkey basters. Those are going into the crus of the penis right here. And I don't know if you can see these tiny little black dots, 1, 2. You guys see those tiny, tiny little black dots right there? That's the exit for these little bulbs. That's the last of the glands. These are accessory organ glands. And it's called the bulbourethral gland. Bulbo, because they're bulbous, goes into the urethra. Bulbourethral gland. Now, I've had students have a lot better success remembering that it's also known as the Cowper's gland. So if you guys want to answer with bulbourethral or Cowper's, either one works for me. Sound good? Cool. So what's going to happen is during an erection, as the erectile bodies in the penis engorge with blood and the penis rises, that's going to start to put pressure as these erectile bodies engorge. It's going to start putting pressure on the urogenital diaphragm, this piece of muscle that separates the abdominal cavity from the penis. And as pressure gets put on the urogenital diaphragm, it's going to cause this bulbourethral gland to squeeze. And when that happens, the fluid from the bulbourethral gland is going to exit into the membranous urethra right here. Yes, no, maybe so? This is a great location to see the ejaculatory duct, by the way. We'll use this and go through a flow chart real quick in just a second. But yeah, now we got the bulbourethral gland. That fluid is only going to enter this tube during the erection. So this is occurring before ejaculation. Does that make sense? So this is a pre-ejaculatory fluid. It contains a lot of mucus to help lubricate the tip of the penis, but it's also extremely alkaline. Yet another basic material, but this is for a slightly different reason. We need to remember that the urethra is a shared tube for male and female, or not male and female, for urine and reproductive system. I don't know where the hell that came from. This is a shared tube for, yeah, exactly, for urine and semen. So urine is extremely acidic most of the time. So this pre-ejaculatory fluid helps clean the system out to make sure that there's nothing in there to kill it from urine. Make sense? So when we go to our flow chart, well, we got something else going into the system, and that's the Cowper's gland. Spongy urethra, all of this dark blue that you guys see here, the light blue is a slightly different type of erectile body called corpus cavernosum. There are two of them. We'll see them in a couple of pictures in a bit. Probably wait until lab to go through those. But this corpus spongiosum goes all the way down to the tip of the penis. The tip of the penis is called the glans penis. Glands, not gland, glands. I'm going to be making references to the glans penis both between today's lecture and tomorrow's lecture, because this is a shared organ. Both males and females have a glans. In males, it's the glans penis, and in the females, it's going to be a major portion of the clitoris. Because males and females all start with the exact same anatomy, and then hormones are going to come into the system and they're going to cause certain things to turn into certain structures, whereas if you don't have that hormone, it turns into a different, another structure. So I like to make sure that you guys understand these references, that, you know, things like the labia majora, for instance. Well, that's just the scrotum that's fused. So we all start with the exact same anatomical features. It blows my mind. I think it's a pretty amazing thing for people to understand a little bit better. Now, on the lab practical, you're also going to need to be able to look at the prepuce. But the prepuce is just the skin. So if I put a pin here in the tip of the penis and I say, what structure is this? The answer is glans penis. If I put it in the skin on the outside of the penis, then the answer is prepuce. Does that make sense in terms of the differences in the terminology? One is the erectile tissue, the highly arousable tissue, and the other is just the skin. All right, cool, we're doing great. We'll come back. Those are the different portions of the penis. There's a root, which has both the bulbous portion and the crus portion. There's the shaft, and there's the glans. Now, at the very end of the story, and there's the corpora there that you guys saw, but again, I think there's better place for me to teach you guys about that. The very end of the story, If I'm asking you to go all the way through ejaculation in terms of a flow chart, you need to make sure that you add the external urethral orifice for me, please, because otherwise it doesn't go anywhere. So the very exit of all of this, it goes spongy urethra. Spongy urethra goes all the way through the glands of the penis. You can put that off to the side, you know, in terms of brackets, what's going on here. This is going to be all three of those. It's the root, it's the body, and it's the glans penis. But the end of the road is going to be the external urethral orifice. All right, cool. Did that help tie together the flow chart for most of you? Cool. All right. We need to get rolling, so I'm going to get rolling. Rolling. Reproductive summarized. Yeah, oh yeah, this one has everything. So let's get down. We'll come back to the female reproductive system. What I want to do is I want to start talking about gametogenesis. And we're going to focus on male gametogenesis, which is spermatogenesis. And then we'll go back and do a flowchart for the female reproductive system, and then we'll talk about female gametogenesis, which is going to be oogenesis. But the gist behind gametogenesis is that these are sex cells. And if I'm dealing with sex cells, then I'm dealing with meiosis. I did a referral to this at the beginning of today's lecture. Males and females, you're going to learn, have a very similar process in terms of meiosis. The differences are few. There's only a few differences, but they have pretty large impact. Most people, I mean, do you guys remember PMAT? Prophase, metaphase, anaphase, telophase? Probably not really. Good, Rudru, awesome. Most people kind of maybe go, yeah, that tickles my brain somewhere. I don't really remember it. Well, the good news for y'all is you don't need to, okay? I'm not going to force you to go back through all of these different phases of mitosis and meiosis. You're welcome. What we are going to do, just as a quick review, is remind ourselves of the difference between mitosis and meiosis. We also want to remind ourselves of what's going on with mitosis and meiosis. When we have cellular division, what do we actually, why are we dividing? What's going on with these cells? What's being passed on? What information's being passed on? And the answer is DNA. You probably, again, don't necessarily remember this, and that's fine. You're not going to be tested on this again. But if a cell is going to divide, it's got to go through DNA replication first. It's got to replicate its DNA. And then it takes that replicated DNA and it puts it into a new cell. It repackages it. I always forget this number. And I might have even gone through it with you guys earlier this semester. How many miles of DNA are in the human body? Each person has about 60 trillion feet, or about 10 billion miles of DNA. That's something like it can go to the sun and back like 30,000 times. That's how much DNA is packed into your body. How do we get that 10 billion miles of DNA? How do we get that packaged into your body? How can we possibly get that much, that long of chains of information inside of your body? There's only one way to do it, and that's to coil it, to pack it, to just turn it over on itself over and over and over and over and over. We take our dunna, which is right here. There's that double helix, and we start to coil it and coil it and coil it and coil it over and over. That is a really good picture of it. We're coiling it, and we package it as tight as we can. That's what a chromosome is. It's coiled DNA. Again, I'm just trying to simplify the heck out of some really complex information. I know once upon a time you were probably taught this, but it just doesn't stick very well because normally the first time you're taught it, you're taught everything about the DNA. You're talking all about all these histones and coils, and the importance tends to get lost. How are we going to get 10 billion miles of DNA into a person? We got to coil it. And what do we coil it into? Chromosomes. Yes, no, maybe so? Now, chromosomes don't always look like an X. They only look like an X during the process of DNA replication. Most of the time, they're actually separated from one another. But the human body has matching chromosomes. Every cell in your body has matching chromosomes. They're called paired chromosomes. Chromosome 1, 2, 3, 4, 5. Well, chromosome 1, you have 2 of them. Chromosome 2, you have 2 of them. Chromosome 3, you have 2 of them. Everybody on board with this idea? So how many paired chromosomes does every person have? Well, that's not exactly true. How many paired chromosomes in what we consider a normal person? 23. Good. Can everybody write that down, please? There are 23 paired chromosomes. 23 paired. So how many total chromosomes does each person normally have? Perfect. 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 That's the number we're after, right? We're after 23 and we're after 46. So a normal cell, that's what we're going to do, is we're just going to write down this normal cell. Got ourselves our circle because, you know, all cells of the body are circular. It's not true. That's a lie. It's just the easiest way to draw this stuff out with my art minor. And we add our 46 chromosomes. Okay, this is skin, this is heart, this is kidney, this is digestive system, this is neurons, these are glia, these 99.9% of the cells of the body are in this stage, 46 chromosomes, packaged into the nucleus. And when the cells go through cellular division, they want to replicate themselves exactly. So as they divide, they're going to double. I'm not going into that whole thing, right? But we end up doubling the number of chromosomes that are here. So what's 46 times 2? Just a number that's going to come up a couple of times, so I want you guys to be thinking about it. It's 92. So now if this cell divides, and we get 2 cells from this process, we're going to take half of those 92 and put them in here. So that offspring has 46 chromosomes. We're going to put the other half over here. So that offspring has 46 chromosomes. This is called a parent cell. It always has 46 chromosomes, and it's going to have 2 daughter cells. These cells always have 46 chromosomes, and these cells are identical to the parent. The 46 chromosomes haven't been messed with. There shouldn't be any error in the translation process, in the DNA replication process. These daughter cells are identical to the parent cell. Right? If you're making a new heart cell, do you want a different heart cell or do you want the same one back in its place? You got new blood vessels. We need the exact same types of cells in there doing that job, right? We don't want to be messing with changing things as we replicate our skin, so forth, so on. This process right here is called mitosis. This is what most of the cells of the body are doing. They're going through this cellular division where they're replicating themselves through a process of PMAT, a single phase of PMAT that ends up creating two daughter cells that are identical to one another. Prophase, metaphase, anaphase, telophase. By the end, we have two identical daughter cells. Any cell of the body that has 46 chromosomes is known as a diploid cell. Okay, so if it has 46 chromosomes, it's called a diploid or diploid. So what are these daughter cells going to be? They have 46 chromosomes. So what type of cell do we end up calling these? These are also diploid. All right, so now what happens with gametogenesis? Well, in gametogenesis, there's going to be a bunch of phases of mitosis you guys are going to learn about, but at some point, you're going to start with a parent cell with 46 chromosomes. So is this a diploid? What is this? 46 chromosomes. What's this thing called? Sure, it's a diploid. Awesome. But now as this thing divides, what's going to end up happening is it doesn't rip the chromosomes apart the same way it does over here. It's a bizarre format with PMAT is that it's going to have a PMAT, but it's going to be a first phase of PMAT. It ends up holding the chromosomes together. Again, you're not going to be tested on every part of this. The moral of the story is that when you're done with this PMAT1, you're going to have a cell, and the cell that has 96 chromosomes stuck inside of it, basically. Because we've replicated this DNA but we haven't actually split it apart. So where there would normally be 46, now we have 46 that hasn't been ripped apart. It hasn't been combined properly. So in other words, there's 23 and there's 23 that haven't been combined in the same way, where we don't have these 46 pairs anymore. And over here, we're basically going to have 23 and 23. Don't overthink this process. You were probably taught this slightly different in another cellular biology class. I'm just trying to get you guys to an end product. So now what's going to happen? These daughter cells don't have 46 chromosomes technically anymore. These daughter cells are going to have a 23 and a 23. They're a little bit of an in-betweener, but those 23 end up making them what's called a haploid cell, not a diploid. Haploid. Oh, I ran out of space. I hope you guys can read that okay. It's H-A-P-L-O-I-D, haploid. And this is called meiosis 1. So there's two stages to meiosis. This is meiosis 1. Now this is going to go immediately from PMAT1 and it's going to enter one more phase of PMAT. So now it's going to enter a PMAT2. Each one of these goes into PMAT2. Technically, it'd be, I and I, Roman numerals. But the product here is going to be splitting these chromosomes apart, ripping them apart, whereas we normally would not. And the offspring's going to have 23, 23, 23, and 23. This is called meiosis 2, the actual stage. Are these cells going to be diploid or haploid? These are haploid cells. This is a process of meiosis. I really truly think this is the most simple way of describing mitosis versus meiosis. I've been taught this so many different ways. I've been taught it. Remember, I was a student for 17 years, so I was taught this in multiple different classes. It didn't really click for me, the importance of this, until I tried to simplify it down to just the bare nubbins, which is where we're at right now. Mitosis produces two identical daughter cells, whereas meiosis, how many daughter cells are there? 4. Each daughter cell has 23 here. Each daughter cell has 46 here. Again, can you guys see that there's a total of 92 chromosomes at the end of mitosis, and there's a total of 92 chromosomes at the end of meiosis? It's not like we're making new chromosomes. We're just splitting them up. Does that make sense? So instead of pairing the chromosomes together, we rip those chromosomes apart. And during this process, what's going to end up happening is the genetic information is going to cross over in our chromosomes. It's a really cool process called crossing over. Now, when our chromosomes go through this meiosis process, wherever they come into contact with one another, here's a chromosome, wherever they come into contact with one another, the genetic information from one chromosome is going to splice and is going to get put onto another chromosome. So what this does is it completely is like this huge, again, remember, we're talking about 10 billion miles total in the human body. So inside of one cell, we're still talking millions of miles of DNA. So imagine a card deck, a deck of cards that's like a million miles, and then you shuffle it. Are you going to ever get the same shuffle twice? The probability is stupidly low. Does that make sense? The biggest deck of cards you've ever even imagined, shuffle them. Are those cards going to go into the same location every single time that you shuffle that deck? No, it's completely randomized, right? Well, that's the cool thing about the end of meiosis, is that every single daughter cell, all of these haploids are going to be genetically different because of this process called crossing over, this huge shuffling, this deck of cards. Is that clear? So over here we have identical daughter cells, wherever over here we have very genetically distinct. Okay, each one of these is going to be genetically different because of the process of crossing over, which is just the shuffling of that DNA, of those chromosomes, on the DNA on the chromosomes. This is the process of meiosis, and this is why every single spermatozoa and every single oocyte is going to be different from the parent cell. Are there some similarities? Sure. But do we, spit out offspring that are literally identical matches to us? No. Every single offspring is unique. Even if you have identical twins, by the way, identical twins still have unique chromosomes. Anybody in here a twin? You kind of know probably what I'm talking about. Anyone in here know somebody that's a twin? When you first meet them, you're like, man, I'm never gonna get to understand, the difference between these two people. And then you hang out with them for just a little bit, and it's like, there's so many things that are different about these people. They look different, they talk different. Oh, you do? Yeah. All right, cool. So that's what's going on here with this lecture. So hereditary information is all about chromosomes. There are 22 what are called autosomes. 23 paired. 22 of those are called autosomes, and then the last one is your sex chromosome. Autosomes are just, those are functionality. Okay, autosomes, get in the zone, autozone. Nope, get in the zone, it's autosome, right? It's all about what makes you you. It's your phenotype. What color is your hair? What color is your skin? Well, those are the boring ones, in my opinion. How many sodium potassium ATP pumps do you produce? What's going on with your thyroid hormone? You know, those are all genetic things. And they're all determined by your autosomes. Remember, proteins are function. And how your proteins are pieced together determines the functionality of your body. Proteins are encoded for by DNA. DNA is the library for your proteins, which is your function. Do you see that flow of information? How tall are you is determined by what's going on with the proteins in your body, and then those proteins help to determine what's going on with bone composition and connective tissue composition, so forth, so on. Cool, awesome. The sex chromosome, X and a Y, this is going to have some genetic information, but again, the Y is pretty benign. The Y chromosome really only has a couple of functional genes on it, and one of them is one that we're going to learn about in tomorrow's lecture. It's called the SRY gene. Capital S, capital R, capital Y gene. The X chromosome actually has quite a few genes on it. And I'm not going to test you on this, so everybody just kind of pay attention here. How about XXY? Is that a possibility? Absolutely. Okay? There's all sorts of different syndromes where the sex chromosomes get a little wonky. Does it mean these people aren't people? I hope you guys are at least, aware enough in life to know that they are people, just because they have a slightly different number of chromosomes than you are, than the normal, quote unquote, normal person. There are other combinations that get a little bit more complex. Right? Is there an XYY? Yes. Talk about even more complex with 2 Y chromosomes. What about somebody with 47 chromosomes? Are they still people? You better believe it. So that's an additional chromosome. These are some of my favorite people on the face of the planet. If we all were as wonderful and sweet as people with Down syndrome, this place wouldn't be so angry all the time. But that 47th chromosome, that's all it takes in order to get something that's slightly off normal. So that's why it's important that every single time that our sex cells divide, that we end up with 23, 23, 23, 23. Because if we make a small mistake and we have 24 instead of 23, even that can cause something like Down syndrome. Does that make sense? This process is very, very well controlled. It's a very strict process of sexual part of the sexual development. Okay, cool. Let me go a couple more slides and we'll take a break. You guys have learned about the diploids versus the haploids, 23 and 23. You've now seen mitosis a little bit more detail. We have, most of the cells of the body cells are called somatic. So most of the cells of the body go through mitosis, where there's 2 identical diploid daughter cells. And these are completely identical. There's no crossing over of information, genetic information, as they are this. How many daughter cells are there? 4. Are they diploid or are they haploid? Haploid. So each has 23 chromosomes, And they've gone through this process called crossing over, where the genetic information gets shuffled. And what's so cool about that is, of course, if we get 23 chromosomes from mama bear, and we get 23 chromosomes from papa bear, and this genetic information is completely shuffled up. It's not just mom and dad, it's a shuffled mom and a shuffled dad. And we have the spermatozoa that fertilizes that ovum. Well, now how many chromosomes do we have? 23 plus 23. Fertilization, how many chromosomes? A 46 bag of mixed genes. Yes, no, maybe so? And those 46 chromosomes become their somatic cells. Those are now their parent cells that are going to help to differentiate and develop every single cell in their body. Crazy that we have that much information stored in 46 chromosomes, that it can create a human being from scratch. Blows my mind. When we come back from break, we're going to talk about spermatogenesis. We're going to talk about the different stages of mitosis. We're going to enter meiosis. What do we know? We're going to start with diploid cells. We're going to end with haploid cells. Not too crazy. It looks crazy. It's not, especially if you followed along with what we just did. Okie dokies. Let's take a break. We're not going to make a long break today because we got a lot to do. So everybody be back at 10:36. Yes, an odd time. Well, technically an even time. Well, that was the fastest 10 minutes on the face of the planet. I feel like I blinked. That went by so fast. We'll take another break. Let's get through some more stuff, though. Um, what I'm going to show you here is the actual process of spermatogenesis. So spermatogenesis is, of course, going to end up with spermatozoa. But what you'll notice here, I like to do this with my flowcharts. Beginning of the story is one cell. How many cells do you guys see down on the bottom? No, not 46, thank goodness. Definitely not 46. And I'm not asking how many chromosomes, Tariq. So I'm asking how many cells there are. So down on the bottom of this screen down here, there are cells, spermatozoa. So how many spermatozoa are down here? You guys are starting to get it, right? There's 16. We start with 1 and we produce 16. For every single time that a sex cell divides and starts to go through the process of spermatogenesis, there will be 16 spermatozoa produced from that one cell. And this is why hundreds of millions of new spermatozoa are produced every single day, because we're always going through a 16-fold increase in the number of cells being produced. All right, so we're going to start with a cell called a spermatogonium. This is going to be the first cell of the process. So if we're going through some male spermatogenesis, which we are, we're going to start with spermatogonium. Okay, this is a singular word for in essence, this is a stem cell. If you ever heard that word stem cell, I know I've mentioned it maybe a few times this semester, but stem cells are origin cells. Stem cells can continue to reproduce and they can become new cells that aren't themselves. So a spermatogonium is going to duplicate itself. And it's going to become two spermatogonia. Spermatogonia is just plural for spermatogonium. So there's one spermatogonium and another spermatogonium that are identical to the parent. Okay, so is this mitosis or meiosis? Right now, what have we done? If a spermatogonium just duplicated itself, created two identical daughter cells, it's just a process of mitosis. So this is normal. 46 chromosomes becomes 46 chromosomes, identical daughters, sweet. One of those daughters goes back and replaces the parent. It's like, okay, parent, you can kill yourself now. Pretty horrible process. Like literally these cells now are going to go through a process of apoptosis, so they go through self-assisted suicide and the daughter takes over. But we got to make sure that this process is seamless. We got to make sure that every single time spermatozoa are produced, that it's coming from a fresh cell, not an old cell, because we can't have any mistakes. The other daughter cell is going to start to move. And I'll show you what this means in just a bit. One daughter cell moves towards the lumen to produce spermatozoa. I'll show you what this means with a diagram and with a flow chart. But this spermatogonia is going to divide. So it's going to go through another state of mitosis. So how many identical daughter cells will this spermatogonium have? If it's going through a state of mitosis, we got 2. And then that spermatogonia, this one spermatogonium and this one spermatogonium, they're going to go through another step of mitosis. We've gone through 1, 2, 3 different steps of mitosis to this point. But we only have 4 offspring. Why not 6? Because one of the first spermatogonium goes to replace the parent. Does that make sense? So we're going to draw this out. How did I get crooked? Whatever. If we go to draw this out, and we're talking about spermatogenesis, because we are. We're going to start with a spermatogonium. and that spermatogoneum is going to divide. And I'm just going to draw a flow chart. This is going to produce 2 spermatogonea. However, one of these spermatogonea is going to go to replace the parent. So now this spermatogoneum that's left over, that goes through a state of mitosis. And then another state of mitosis. This one up here was mitosis as well. And if I take one cell, divide it, and then divide it, I'm going to end up with 4 cells. True story? You guys can write this out however you want to make sure that you remember the numbers. I like to put as little as possible in my flowcharts. It forces me to learn the portions of the flowcharts that are missing. Now, for what? So we're going to change this thing's name. It's going to be no longer called a spermatogoneum. See, at this point, we're going to enter meiosis. Right down below this, we're going to enter meiosis 1. So because we've entered meiosis, this is no longer called a spermatogoneum. This is now going to be called a spermatocyte. So now this is going to become a cell that becomes a sperm. So we call it a spermatocyte. And since it's entered meiosis 1, we call it a primary. Does that make sense? So that's the terminology. We're going to have 4 spermatocytes. And these are going to enter. I didn't leave myself enough room. Dang it. These are going to enter meiosis. That's what's happened right here. These spermatocytes enter meiosis 1. Therefore, we call them primary. I'm just going to end up having to write that over here on the left. Sorry about that. Because they're entering meiosis, Melissa. They're not going through mitosis. If they were to continue mitosis, we'd end up with identical spermatogonea. But we're changing the process here. The spermatogonea is no longer going to go through mitosis. It's going to go through meiosis. So we're going to get rid of that 4 right there. We're going to say primary. Because it's meiosis 1, it's primary. And yeah, there's 4 of these. All right. So remember what we did with meiosis 1 before is we said basically that at the end of meiosis 1, we've gone through one phase of PMAT. We've duplicated our DNA, but we haven't ripped that DNA apart yet. Yes, no, maybe so? So these primary spermatocytes are going to enter meiosis 1 and they're going to complete meiosis 1. At which point they're going to enter meiosis 2. You guys with me? Come on, computer. So if we've finished meiosis 1, now each one of the primary spermatocytes has duplicated itself. And each one of these is now going to be considered a haploid because we got 23 and 23. They're not fused together and they're not ripped apart. So we got 23 individual chromosomes, 23 individual chromosomes, 23, 23, 23, 23. They're in here as separate entities. These are now going to be entering a second phase of meiosis, so we call it a secondary spermatocyte. And how many are there? 4 just divided, therefore there is 8. Yes, no, maybe so? And these are secondary spermatocytes because they're entering meiosis 2. Now, at the end of meiosis 2, meiosis is done. right? And these cells are going to duplicate again. So now here we go. This is meiosis 2. I'll actually go ahead and write these in with my arrows too. I like to do that, so I'm always aware of what I'm talking about. Meiosis 1 is complete, and immediately meiosis 2 starts. And when meiosis 2 is done, we've gone through that final phase of PMAT2, and now the chromosomes get ripped apart. Okay, that means we're going to duplicate again. So how many are going to be going on here? How many cells do we have? And that's where we get our 16, right? Now these are going to be called spermatids. We're no longer spermacytes. We finished meiosis. So we're no longer spermatocytes. We are now spermatids. Okay, we're done with meiosis. And now they're going to go through a process of getting packaged and getting mobilized. It's a maturation process, although they're still not mature spermatozoa by any means. So these cells are going to start to grow tails and all that craziness. These spermatids are basically, if you go through like a frog phase, we start with the frog egg, that's the spermatid, and then that little frog turns into a tadpole, and that tadpole would be the spermatozoa. So these spermatids, they're done with mitosis, they're done with meiosis, they're not dividing anymore. Now they change. Yes, no? And they get packaged. They go through a process of kind of almost differentiation. They're getting packaged into spermatozoa. And since we're not dividing anymore, there's just 16 of those. Okay, no more mitosis, no more meiosis. Now we have 16 spermatozoa. Each one of those with 23 chromosomes. 46, 46, 46. Every time we go through mitosis, there's 46. But when we enter meiosis, now there's 23. Does that make sense? So let's cluster these together. Spermatogeneum. I'm going to go ahead and call this step 1. Bear with me. This is going to work with the anatomy. So I got step 1 right here. The spermatocytes, we're going to go ahead and actually, let's do spermatogeneum as step 1. Spermatogenia, as it's dividing, is step 2. And now those primary spermatocytes are going to be step 3. Secondary spermatocytes, those are step 4. Spermatids, those are step 5. And the spermatozoa, that's going to be a step 6. We're just going to cluster each one of these mitotic stages up here. As the spermatogenia go through mitosis, we're going to just cluster all that together. All right, where does spermatogenesis occur? Where does spermatogenesis occur? Yep, you got it. See? The little bit of memory that works, right? Those funny little jokes, they work, they stick. Seminiferous tubules. So let's take and look at a cross section of a seminiferous tubule. Just like a blood vessel, if we were to take and cut a tube, you're just going to see a big round circle. Yes, no, maybe so? Again, my art minor with that perfect circle. I'm never going to let down the art minor thing, by the way. I'm going to be telling that story for the rest of my teaching days. Thanks, Dreek. I got a good laugh out of it, and I'm going to continue to laugh about it. Now, what's going to happen in this tube is we're producing new spermatozoa. And it all starts from out here, and it's going to move towards the center of the tube. So let me use a different color. So we're going to start from here, and we're going to be moving towards the center of the tube. And it's just a flow of information. One is way over here next to the outer border of one seminiferous tubule. So this is where step one is going to occur. So what's going to happen next, y'all? Step two, what's going to happen next? Step three, what's going to happen next? Four, what's going to happen next? Five, what's going to happen next? Six. Does that make sense? And what we end up with is a group of cells between the outer border of the seminiferous tubule and then the lumen on the inside. Once we make it to the lumen, that's actually where 6 is going to be. I probably should have drawn that inside of here, but that's where we're going to end up with our spermatozoa. But in the interim, we're going to be running along 1, 2, 3, 4, 5. As these cells divide, those cells take up space in that seminiferous tubule. Let me show you guys a picture. I just like to get you guys having a reference. But if I take a single seminiferous tubule, this has a lot of crazy stuff going on, right? Ignore all that crazy stuff. Ignore all the words. Ignore everything. And you'll notice that I have a tube. Here's the outer border of that tube. Here's the inside of that tube. Yes, no, maybe so? So what did you guys learn? The large majority of the cells that are going to be along the border here, are they going to be spermatozoa or are they going to be spermatogonea? Good. Those are going to be spermatogonea. And as they divide, they're pushing out towards the center of the tube. Now, what about here in the middle of the tube? What are these funny looking tadpoles going on here? What's living here inside the lumen, getting pushed around by cilia? What's here? Does that make sense in terms of our little, yeah, I mean, I guess our clock here, right? We went from 1, divides, pushing out, 3, 4, 5. So if I wanted to try to find my spermatids, where am I going to look? Am I going to look over here next to the border? Or am I going to look over here closer to the lumen of the tube? The answer is obviously closer to the lumen of the tube. It's going to be this border right along the edge. You can see there's a whole bunch of smaller kind of anuclear because we're no longer in these states of mitosis anymore, and these are the spermatids. Make sense? You are not going to need to go through you are not going to need to identify spermatids, spermatogonea, or anything along those lines on histo. That's not the point of this. The point of this is to see that it's a flow of information, just like everything else in the body. And that is going from structure to structure to structure to structure to structure. Is everybody clear on that? Lovely. So there's something else going on here in the seminiferous tubule, a really important structure that's going to help determine the function, but you're not going to see it under histo very well. So we got to go to a depiction, right? An animation or whatever these things are called. And what you'll notice here is here's that flow of information. We're along the outer edge of the seminiferous tubule. Don't read all these words. Please just follow me. This is something slightly different. We'll come back to this. So there's that spermatogoneum. It's going to go and divide and divide and divide. There's going to divide into primary spermatocytes entering meiosis 1, secondary spermatocytes entering meiosis 2. Meiosis 2 is complete. We get spermatids, and now they start to package, and we get spermatozoa out in the lumen of the tube. Does everybody feel comfortable with that flow of information now to some degree or the other? The more you see it, the easier it'll become. Now, what you'll notice is that the cells, spermatocytes and the spermatids and stuff, they're not the only cells here. There's actually this big, huge orange blob. and this big, huge orange blob that is going all the way from the border to the lumen. There are these huge cells that take up this entire space, and you can't differentiate them in the histo because you can't see the cell boundaries. These big cells, and this process of spermatogenesis is occurring through them. It's actually going between them. One of these cells and one of these cells, spermatogenesis is occurring between them. Okay? These cells are called Sertoli cells. That's the word that I want you guys to know them as. However, you might in your homework see them as sustentacular cells because they help to sustain spermatogenesis. You might also see them named nursing cells because they nurse spermatogenesis. You might also see them called columnar cells because they look like huge columns. Sorry, it's not my fault. Here, they're called sustentacular cells in this diagram right here. And you'll notice that in this picture, there are these nuclei that are very columnar in shape. Right here, there's one. Right here, there's one. Right here, there's one. There's a smaller one. There's one. Yes, no, maybe so. You guys see these big columnar? They're oval, but when you're dealing with the cell, if you have this big, long, skinny nucleus, that's part of a columnar cell. Those are going to be those are the nuclei of your sustentacular cells. But again, I want you guys to know those as Sertoli cells. And these Sertoli cells are going to be a big part of your exam. They are a very important cell in this process because it's these Sertoli cells that allow for spermatogenesis to even occur. Without Sertoli cells, no spermatogenesis. And they do so many other things. Just like there's a blood-brain barrier, there's a blood-testes barrier. In the brain, that blood-brain barrier is created by astrocytes. These are basically the astrocytes of the testes. We want to make sure that no pathogens, no viruses, no bacteria get down and mess with the process of spermatogenesis. So we create a super ultra filtrate, and we get the nutrients, and only the nutrients that those spermatozoa need to undergo division and this immature maturation process that's going to be going on here. It's these Sertoli cells that provide both the barrier and the nutrients. These are also extremely phagocytic cells, so they're part of your immune system. If something foreign gets here and tries to mess with the spermatozoa, these Sertoli cells are going to freak out and they're going to eat it. They're going to engulf it and they're going to digest it. Bacteria, virus, whatever the case may be. These use that ultrafiltrate of the blood and they start to secrete seminiferous tubule fluid. This is the fluid that allows for these immature spermatozoa to start passing through the epididymis, up through the vas deferens as it follows and is, creating the cilia of that, those tubules are going to be creating currents for that fluid. These Sertoli cells secrete a crazy important blood protein. What do we know about steroids? Are they polar or non-polar? And if they're non-polar, can they just dissolve in your bloodstream all happy-go-lucky? No. If they're non-polar, heck no. Right? If they're non-polar, do oil and water mix? No. So if it's non-polar, what does it require in order to be carried through the blood? A carrier protein. not a transport carrier. You're just combining your terms, Ashley, not a problem. There are transport proteins that transport things across the cell membrane, and then there are carrier proteins that carry them through the bloodstream. Yes, no, maybe so? Everybody on board with this idea? All right. Testosterone is androgen. Androgen is testosterone. So androgens are a base steroid that all of your sex hormones are produced from. So estrogen, progesterone, testosterone, these are all a form of androgen. So androgen is a term for a steroid hormone. So now you guys can start to connect the dots. These Sertoli cells produce ABP, androgen binding protein. This is the Uber, and since this is the male reproductive system, what is this an Uber for? Testosterone. Without the proper secretion of ABP, testosterone's not going to get out to the rest of the body. That male's bone's not going to grow correctly. Muscle development is going to be greatly inhibited. Energy, so forth, so on. They could be producing 4 times the amount of testosterone as the normal male, but it doesn't matter. If it can't be carried through the bloodstream, then that male doesn't get that testosterone. Does that make sense? These Sertoli cells are also target cells. What are they a target cell for? They have receptors for testosterone and follicle stimulating hormone. So they're target cells for testosterone and FSH. You're going to see this in some flowcharts here, but the reason why is because testosterone and FSH are required for spermatogenesis. Follicle stimulating hormone was first found in females and helped with follicular development, but here in males, they don't have follicles, they have spermatozoa. We found that it's the exact same hormone that the females have, so we didn't want to rename it, so we still just called it follicle stimulating hormone. But instead of stimulating follicles, now it stimulates spermatogenesis. What cells are required for proper spermatogenesis, everybody? What cells in the male are required for proper spermatogenesis? Sertoli cells. There's one more hormone here, bold red. You can see that I definitely want you guys to know these things, testosterone, FSH. And then this guy right here is called inhibin. This is a hormone. It gets into the bloodstream. Inhibin is a hormone. It gets into the bloodstream. What do you guys think inhibin does? Is it going to activate something or is it going to inhibit something? It's going to inhibit something, and it's a pretty important inhibiting hormone. Otherwise, they wouldn't end it in, it's IN because it's an active protein. That's why we end things in IN, and they wouldn't have named it inhibin otherwise. What does an inhibit? It inhibits FSH. Oh, you definitely, this is not as simple as it. The only reason why this is simple for y'all is because you have a pretty extensive background now. Just imagine, the word inhibit before you even entered A&P. You probably didn't even know what that word meant, right? So I love this idea of you guys learning, and things just get more and more simple as you learn. That's the great thing of learning. If you've really put your time and effort into it, then the next thing you learn gets easier. We did not talk about inhibin in the renal system. No. We talked about secretin, motilin, but not inhibin. All right. So inhibin is secreted by Sertoli cells, and it's actually going to go to the source. We're going to draw some flowcharts in just a bit, and it's going to block FSH. So I'll remind you guys where FSH is secreted from. Inhibin is going to have receptors up there, and it's going to block FSH secretion. Inhibin is a very specific inhibitor of FSH. Please write that down. Put it someplace special. And then we're going to draw some flowcharts, and we'll make it even more special. All right, I'm moving past female reproductive system. Move in, move in, move in. And we're going to start talking about the male spermatogenesis and the controls for male spermatogenesis. We're going to draw a flow chart for this. Who remembers from Unit 1 that the brain actually controls spermatogenesis? Probably not a lot of you, and that's okay. Not me. Good. Honesty's good. Right? But let me start off with a flow chart that maybe you remember. Might cause a little bit of PTSD, so hold on to your hats. You guys remember this? Oh, yeah, inulin. We covered this pretty extensively where we talked about hypothalamic hormones, talking to anterior pituitary hormones, which now can communicate with target cells. One of those hypothalamic hormones, I said, let's not worry about this in Unit 1. We're going to talk about it in Unit 4. Here we are. That hormone was called gonadotropin releasing hormone. Gonadotropin, where tropin means it's a hormone that causes the release of another hormone. Gonad, as in gonad, right? So it's going to be talking to the gonad eventually, releasing hormone because it's getting secreted from the hypothalamus. And that gonadotropin releasing hormone goes into that portal system. It goes down to that anterior pituitary, and it's going to cause the release of 2 different hormones. Anybody remember now? What are the name of those 2 hormones secreted from the anterior pituitary? Yes, and yes. Is this ringing a bell? We had FSH, and we have LH. We have follicle stimulating hormone, and we have luteinizing hormone. Yes, no, maybe so? Awesome. I forgot to mention something, and I just realized that. Let's go all the way back to this picture, way back here. Yeah, it's not showing there, it's showing here. There's an important cell that we did not discuss in this histo. Again, you're not going to have to identify, that's not true. You will have to identify it for histo. I lied, you will. There's a cell that does not belong to the seminiferous tubule. This is the tube right here. But what you'll notice is that there's cells that live outside of the tube too. Yes, no, maybe so? There's cells out here. That's not a tube. These are known as interstitial cells. Now, you're not going to learn them as interstitial cells. I'm not going to let you. I'm going to make sure that you guys know them as their proper name, which is Leydig cells. L-E-Y-D-I-G, Leydig cells. Sometimes it's nice to just look at these little diagrams before you go any further. Here's a seminiferous tubule, where the spermatogonea are moving towards the lumen with the help of Sertoli cells. So Sertoli cells are found inside of that seminiferous tubule, but in between, there's a lot of space. Maybe not a lot, but it's there. And that's where we find these Leydig cells, otherwise known as interstitial cells. So this is a pretty good picture for it. You can see a seminiferous tubule, seminiferous tubule, seminiferous tubule, and there's cells that live in between in between, there's a nice little patch of them right there. These are your Leydig cells or your interstitial cells. If I zoom in a little bit more, here's one seminiferous tubule, there's another, there's another, there's another, and there's cells that live in between. Yes, no, maybe so? Everybody feel comfortable that they might be able to identify these cells? Leydig? So why are they important? Because Leydig cells secrete testosterone. Everybody, what hormone do Leydig cells secrete? Do females have Leydig cells? No. No, definitely not. Because females aren't going to be producing bucket loads of testosterone, right? These interstitial cells, these Leydig cells, these are the ones that secrete the buckets of testosterone required to turn off a male's brain. I mean, with male growth. Yeah, they definitely produce testosterone, but where is their testosterone coming from? Not the gonad. They have testosterone coming from the adrenal gland. You guys learn about that also in Unit 1. The adrenal gland has the adrenal cortex, GFR, glomerulosa, fasciculata, reticularis. Yes, no, maybe so? Glomerulosa is what secretes aldosterone. That fasciculata is where cortisol came from. Now that reticularis, the deepest portion of that adrenal cortex, that's always producing sex hormones. So in both males and females, we're getting estrogen and testosterone. But the difference, of course, is that males have Leydig cells. They have a gonad that's producing a lot of testosterone. When females, they have their gonad. They have an ovary that during cycles is going to be producing lots of estrogen. All right, you guys with me? Did that make sense? So those Leydig cells are important. All right, cool. Let's get back down to the fun stuff. The brain directs reproduction. This can't be understated, is that ultimately the brain controls pretty much everything, of course. Whether it's through nervous control or endocrine control. Actually, this leads into the segue you guys were asking about, right? Sex steroids are closely related to one another. Whether or not we're talking about testosterone, androgen is testosterone, or byproducts of androgen. As androgen goes through, biochemical reactions, it becomes estrogen and progesterone and so forth, so on. Now, remember, both males and females have these testosterone and estrogen being produced from the adrenal cortex, which is what we just talked about, which is down here in this bullet point. Now, the reason why testosterone is dominant in males is because of what cells? So go ahead and answer that in chat. What cells in the male are constantly producing bucket loads of testosterone? Great, awesome. And then the ovaries are producing the estrogens, estradiol, estrone, progesterone. And by the way, they produce even more androgens. You guys are going to see these androgens are required to make their estrogen. So we'll get into that more soon. So let's go back to here because I love doing these little reminder flowchart things. I tend to get a connection with students' memories when I do it this way. So what do we know? What do we know about the receptors for FSH? What did we just say? Where do we even find these receptors for FSH? We just had a slide on it not too long ago. Yes, those are those Sertoli cells. So if I'm going to say where the target cell's at, target cell is a target cell because it has receptors, well, that is Sertoli. Does that make sense? And now Sertoli cells, they cause a function, which is spermatogenesis. Lovely. Sertoli cells also secreted a hormone that acts via negative feedback. What hormone did Sertoli cells secrete that directly inhibits FSH? So now we got inhibin being secreted by Sertoli cells, which will directly inhibit FSH. So what do we now start to see? If Sertoli cells are overly activated, spermatogenesis is going through the roof. Then Sertoli cells are going to start to secrete more inhibin, which directly inhibits FSH, which is going to do what to spermatogenesis? Increase, decrease, no change? Homeostasis, right? We're constantly trying to control the body via homeostasis, even something like spermatogenesis. Everybody feel pretty good with what we just did right there? So let's go LH route. LH, the receptors for LH are somewhere else. You can use your critical thinking skills. Where the heck are the receptors for LH going to be? What other major cell of the seminiferous tubule did we discuss? About 3 minutes ago. Starts with the letter L. So that's why it was important that I went back and reminded you guys or taught you guys about Leydig cells. Yes, no, maybe so? It's because the receptors for LH are found on the Leydig cells. And what is the function of Leydig cells? What do they secrete? And where do we find receptors for testosterone? On those Sertoli cells. And it helps with spermatogenesis. Yes, no, maybe so? We saw that in a slide not too long ago. Now we're tying it all into one big flow chart. Now, where else do we find receptors for testosterone? Up in the hypothalamus and in the anterior pituitary. And the function of these receptors is, you probably guessed it, what's going to happen if testosterone levels get too high? What's the body going to try to do? Increase, decrease, no change, testosterone levels. This is negative feedback. As testosterone levels get too high, the anterior pituitary will inhibit the secretion of FSH and LH. If testosterone levels get too high, the hypothalamus is going to slow down its secretion of GnRH. And what's going to happen to spermatogenesis if there's too much testosterone in the system? Increase, decrease, no change. Got it. It's counterintuitive. A lot of people think that if you have raging testosterone, that you're going to be producing tons of sperm. What did you just learn? Is that true? Nope. Can you still be sexually aroused? Of course, right? Is there still going to be a virility that's going on? Absolutely. But are you going to be sterile? Most likely. Where this comes into play are roid users. So people that take exogenous testosterone levels. Anybody ever heard the rumor that they are sterile? Tiny little gonads? Well, it's not a rumor, it's true. Because as you take exogenous testosterone, you're bypassing your entire system. You keep pumping testosterone into your system and you get do big muscles. Which way to the gym. But the body is going to say too much testosterone, shut down. So it's going to stop Sertoli function. It's going to stop Leydig function. Spermatogenesis stops or decreases to the point where they're basically infertile. Get it, got it good? I love physiology. It's so cool. What hormone is secreted by the hypothalamus to promote spermatogenesis. Just getting you guys to do some redundancy here. GnRH. What hormone is secreted by the anterior pituitary that communicates with the Sertoli cells to promote spermatogenesis? What hormone is produced by and secreted by the anterior pituitary that promotes testosterone release from Leydig cells? Where do we find receptors for testosterone? All over the body. But where do we find receptors for testosterone that helps with spermatogenesis? Sertoli cells. If Sertoli cells are too activated, testosterone levels are high, spermatogenesis levels are high, what hormone will Sertoli cells secrete to inhibit FSH? And that's going to help shut down spermatogenesis. You shouldn't have to write any notes here. Hypothalamus secretes GnRH into the portal system, travels the centimeters down to the anterior pituitary, causes secretion of FSH and LH. FSH goes into the bloodstream, and the receptors for FSH are found on the Sertoli cells. Sertoli cells now have a number of functions, one of which is to promote spermatogenesis, and it's going to cause a spermatogonium to go through process of mitosis and meiosis, and we get more sperm. In addition, when spermatogenesis levels start to get too high, these Sertoli cells produce inhibin, which now goes back to directly inhibit FSH. In addition, something we didn't add to our flowchart in our Chalk Talk is that Sertoli cells are going to start to secrete more and more ABP, androgen binding protein, which is designed to bind testosterone. Why? Well, because we can expect that testosterone levels are going to start to go up. Because what's going on with LH? LH is secreted in the bloodstream. The receptors for LH are on the Leydig cells. And the Leydig cells start to produce more testosterone. That testosterone goes into the bloodstream. Well, boom, ABP Sertoli cells, remember these Leydig cells are just outside. They're in the interstitial space right here. So now that testosterone, which is nonpolar, just fuses out into that interstitial space, into the blood supply, where it binds to ABP. In addition, those Sertoli cells have receptors for the testosterone, and it helps promote spermatogenesis. But if testosterone levels get too high, good news. We have hypothalamic receptors and anterior pituitary receptors for testosterone, and it's going to inhibit both of those systems. Testosterone is also really important for basically all other male growth functions, so growth hormone, thyroid hormone, even epinephrine action. In the male, a lot of that is going to be synergistic with testosterone that's available. Do you guys feel pretty comfortable with this flowchart? Is there any part of the flowchart you need me to cover again? Might just be a matter of you sitting down and, writing it out a couple of times from scratch for yourself. In essence, it's the exact same thing we did right here. This one's just got, pretty pictures. All right, let's take another short break. Everybody be back. Oh, how ironic. 36 after again. We are done with male repro. Crazy to think about. Went pretty fast. Helps if I unmute. Ashley, kind of a semantics thing. But if you look at this picture right here, seminiferous tubules winding back and forth, top ramen. Inside here, the tunica albuginea is this light blue. And the tunica vaginalis is, you can think of it, again, parietal layer and a visceral layer. So just like you saw in the Bowman's capsule, and just like most of the rest of the serous membranes of the body, there are two layers, parietal layer and a visceral layer, and in between that's a cavity. Helps protect, lots of fluid, yada, yada, yada. You guys probably get the point of that by now. So that's the tunica vaginalis. It's a combination of that parietal and that visceral layer. So in terms of the word housed, yeah, sure. Right? That's the outer shell of the house, and all of this stuff lives inside of it. Does that make sense? You're welcome. Okay. Female repro. I want to try to go even faster. Female reproductive system is more complicated. Not necessarily the organs. The organs themselves are, it's the same tissues, right? All these primary sex organs and accessory sex organs in the female reproductive system, they are derived from the exact same tissues as the male's sex organs are. So the gonad, female gonad, is the ovary, and the accessory sex organs are everything else. Right there, that little white ball that you see right there kind of looks a little bit like the tunica albuginea of the testes, doesn't it? Because there's a similarity there. It all actually starts with the same tissue. And that right there is the ovary. All right, so we can look at it from the side view, and we can look at it from the front view, and it still kind of looks like that tunica albuginea. Now the inside looks very different. The inside does not have seminiferous tubules. Instead, it's got a whole bunch of different, what's called follicles, in different follicular stages. And that's going to be one of the complex parts of the female reproductive system. We're going to deal with that in a little bit, and I'm not going to do it now. But the analogy that a lot of people like to use in the textbooks and stuff say that this looks like a bighorn sheep. Right, so when you're looking at this system, there's that bighorn sheep nose, and there's the big horns. You guys know, I guess not everybody always knows what a bighorn sheep looks like. I once was catching bighorn sheep for a brucellosis study. We were swabbing their noses, and oh, come on, internet. What's going on here? Right, so there it is right there. So you can see the curls, and there's the, there's the uterus, there's the cervix. We were doing these brucellosis nose swab studies, and this bighorn sheep, this big male, turned around, and instead of going into the net, the people that were pulling on the net while I was chasing this bighorn sheep, they pulled it too early, so the sheep saw it, and it turned around and it rammed me right in the chest. Hurt so bad. Anywho, still caught it. I grabbed my arms around its horn and brought it to the ground. But it hurt. So there's that bighorn sheep head. Now, if you listen to my narrated PowerPoints and or even my YouTube lectures on this stuff, I don't really like that analogy all that much. The analogy I like is just a person that's holding two footballs. This analogy works better for me. Just imagine your arms out to either side, kind of holding it up almost like Karate Kid style, but now you got two footballs in either hand. Why that's important is because look at the anatomy. Do you guys see these big long tendrils? These are called fimbriae, which means fingers. And here's the football. It's the ovary. Don't fumble that football, right? Yep. I'm waiting. I should be back now. Am I back? Hi. I don't know why my computer just rebooted the entire Blackboard, but it did. But the analogy goes, if you're holding two footballs up, is or are those footballs directly connected to your hands? No. You're holding on to those footballs. Does that make sense? It's not fused to your hands. You're just holding them there. And the ovary is a little bit creepy like that. The ovary is not connected to anything else except for some ligaments holding it in place. And the ovaries is where all this follicular development goes on. And the ovary is what produces the oocyte through the process of oogenesis. And the ovary is what's going to ovulate that oocyte out in the middle of nowhere. And you can kind of see it here. You guys see that tiny little oocyte getting popped like a zit? Literally is like a really nasty, horrible cysty zit. Massive amounts of zitty fluid coming out right here. Ooh. And it just gets erupted out into space, out into nowhere. This is the peritoneum. This is nothing. And the female body's got to take care of the rest. So what's going to end up happening is the fingers of the fallopian tube. This is the uterine tube, also known as the fallopian tube. I like to call it the fallopian tube. You're more than welcome to call it the uterine tube. The fingers, the fimbriae, when these hormones are changing in the ovary and ovulation occurs, the fimbriae start to move. You can imagine your fimbriae are your fingers, and then you can imagine, I don't have a football near me, but now imagine that your fingers are starting to move. That's going to create a fluid current. Yes, no, maybe so, as those fimbriae are moving or creating a current. So now this fallopian tube has a lumen. It's got an opening to the straw. The very end, the very opening of that straw is called the infundibulum. We've seen a couple of infundibulum. This infundibulum is of the fallopian tube. So if I were to ask you what specific structure is this, you would say infundibulum of the fallopian tube. Exactly. There's an infundibulum that connects to the pituitary gland. But this is the infundibulum of the fallopian tube, okay? And the current that has been created by the moving fimbriae is going to cause that ovulated oocyte, hopefully, to get sucked up into a vacuum, into that fallopian tube. Is it going to always work? Hopefully. But in a lot of women, their fimbriae, their ovary, maybe their ligaments are slightly out of shape, so their ovary is not perfectly in line with their fimbriae, and when ovulation occurs, they ovulate out here. And that fertilized oocyte does not even get to the fallopian tube. Kind of hard to fertilize then, huh? Lots of things can go wrong. I always make jokes about this, but everybody took like a health class probably when they were a kid, and they're like, don't have sex because you will get, let's do a Mean Girls quote, you will get chlamydia and you will become pregnant and die. Right? It's like the second you have sex, They teach you that you're going to get pregnant. It's not that easy. You know, some people, my wife and I tried for like 6 years. So it's not always easy. Lots of extenuating circumstances, but it's because the physiology behind it and the anatomy behind it is not easy. As that mature oocyte enters the infundibulum, it goes to the ampulla of the fallopian tube. We've seen a lot of ampulla this semester. This is just another one. Don't just write ampulla. You got to write ampulla of the fallopian tube. And just like the male ampulla of the vas deferens, which stored mature spermatozoa, well, now we got an ampulla of the fallopian tube. That is where this mature oocyte embeds. It's here that it stops its journey. A lot of people don't realize this. Every semester I get people that are pretty surprised that this is where fertilization occurs. It does not occur in the uterus. Okay, those spermatozoa are going to have to go into the vaginal cavity, past this booby trap right here, into that cervical canal, up into the uterus, war zone, nuclear bombs, all the way through. They're going to have to swim you got to find this tiny little narrow passage all the way up into the ampulla in order to fertilize that egg. It is a journey and a half. Only the strongest should survive. Oh man, it's nasty in here. And in a good way. Flow chart. You guys see it? Ovary, ovulation, fimbriae of the fallopian tube, infundibulum of the fallopian tube, ampulla of the fallopian tube. If you want to go with fertilization, we wait for fertilization. And now, once that egg is fertilized, that fertilized egg is going to travel down the fallopian tube. There's quite a few different portions of the fallopian tube. There's a body, there's an isthmus. Isthmus just means narrowing. So there's a narrow neck of the tube that now enters into the uterus. The uterus itself has multiple different regions and different layers. So we'll get into that in just a second. But that fertilized egg is now going to embed itself into the uterine wall, and hopefully it goes through the proper process of fetal development. But it doesn't always work. This is a tough job. So if the uterus isn't perfect, if that wall's not perfect, if that fetus is developing improperly, the body's going to recognize this, and it's going to hopefully, because it can be very dangerous to have improperly developing fetus growing inside that uterus. By the way, what would happen if the fallopian tube didn't properly transport that fertilized egg? What if it accidentally got embedded here? We have a fertilized egg. Now we're dealing with an ectopic pregnancy. You got it, fallopian, which is a type of ectopic. Very dangerous. Very, very dangerous to both the mom and, of course, the baby's not going to make it. So a lot of times they're going to have to go in and do, surgery. Sometimes they'll have to do a hysterectomy. It's pretty brutal. So yeah, lots can go wrong during pregnancy. Now, let's just say that baby is developed normally. We're, exactly 40 weeks, because we know it always works like that, right? Exactly 40 weeks. The baby is now perfect. It's going to say, Mama, I want out. It's going to knock on the door, and then it just slides out like a water slide. Yeah, that doesn't work like that. But as that pregnancy, that birth is occurring, that baby is going to be traveling through the cervix. Now, the cervix is this entire area down here. All of this, cervix just means neck. So this is the neck of the uterus. So this is the cervix. Now the cervix has a cervical canal and an opening on the inside and an opening on the outside. So as we are leaving this system, it's going to go internal os through the cervical canal to the external os. Finally, it goes down to the vaginal canal, and we'll talk more about the vaginal canal. We'll even talk about these fornix up here because you know there's some funniness that goes on with those the fornices. We'll talk more about the vagina later, but kind of everybody get a pretty good idea of the flow of information here. Let's take a blank one. Ovary, fimbriae of the fallopian tube, infundibulum of the fallopian tube, ampulla of the fallopian tube, body of the fallopian tube to the isthmus, into the uterus. Now, the uterus has a number of different regions. One of them should be familiar to you. Just like the top of the stomach, we call this the fundus. Just like the main portion of the stomach, we call this the body. Now, instead of having a pylorus, we have a cervix. Okay, so the regions of the stomach shouldn't be too complex for you. Did that make sense? Fundus, body, cervix. That area is called the ampulla because it is a bunch of so this picture doesn't show the individual ampullae. That's part of the reason maybe you're confused. But remember, an ampulla, like if we were talking about an ampulla as a vial, like people give patients medicine in ampulla. Okay, and an ampulla well, it's like a tiny little, well, gosh, man, it pulls up all sorts of anatomy because that's what I look up most of the time. But if I'm looking at a vial, right, like you give like a little ampulla of sterile vacuum, I don't know, whatever the hell was in here. But yeah, does that make sense? Like each one of these, like these little glass ones that you break the top of, that's called an ampule. So they're just tiny little individual storage containers. And in the human body, those ampulla are tiny little individual storage containers. Well, we have these kind of areas. It's not quite the same as it is with the vas deferens. It's not quite as distinguished of ampulla here, but there are openings here that help to trap that ovulated egg. All right, cool. Fundus, body, cervix. Cervix, you guys have already seen, has a internal os, a cervical canal, and an external os. The next part of the anatomy that I want to talk about is that the uterus has layers, just like ogres, right? There's always layers. We've seen this through the entire body. Now, the layers of the uterus aren't too complicated. You've seen this through so many systems to this point. We got an outside, we got a huge muscle, and we got an inside. So I like to think of it like the heart. Remember in the heart, we had a pericardium. We had a muscular layer of the cardium, which was myocardium. And we had the endothelial layer, which was the endocardium. Yes, no, maybe so? Peri or epi, just depends on which organ we're talking about. We saw this with bone, periosteum, endosteum. We saw this with cartilage, perichondrium. We saw this with even neurons, for Pete's sake. Perineurium, epineurium, endoneurium. So we've seen this context over and over and over. Well, here in the uterus, I'm going to steal this arrow because I'm not sure I have it. We get these same three layers. We get a peri, we get a myo, and we get an endo. But all we have to do, if it ends in IUM, right, that means membrane or layer. So we've seen that repetitively. But now we got to throw in metria. Metria means uterus. perimetrium, myometrium, endometrium. So far, so good? Just because I like to put in real life to what you're studying, and I like to put in things that a lot of people are familiar with. How about endometriosis? Has any of you ladies here had endometriosis or know somebody that has? That's from the term endometria. Endometria is that inner layer of the uterus. And the endometrium actually has a number of different layers to it. The endometrium's not super simple, but we're going to try to simplify it. We're going to break it into two layers. One's called a basal layer, a stratum basal, so to speak, right? It's a basal layer of the endometrium, and then there's a functional layer of the endometrium. Now, the basal layer of the endometrium is there every day of your life. It doesn't change. The basal layer is just the normal layer of endometrium that women have lining their uterus. It's basal. It's a basement. It's what's attached to the myometrium. Right down here is the muscle. So what the hell is this? This is the functional layer, the stratum functionalis. What's going on up here? This is the layer of the endometrium that regrows itself every month. This is what's happening during the uterine cycle, portion of the menstrual cycle, is that as you are going through both ovarian cycle and uterine cycle, the uterus is building up the uterine wall, preparing that uterine wall with brand new blood vessels and brand new glands every single cycle. We want everything to be perfect when implantation occurs. We can't wait 2 months. We have to make sure that this is a new wall every time. You guys see the difference between the basal layer and the functional layers of the endometrium? So what's happening here in endometriosis? This is where endometrial tissue starts to grow where it shouldn't grow. That uterine lining makes it out of the fallopian tube, and it's a very powerful, highly mitotic cell line. And it causes massive irritation, inflammation, and pain. Crazy amount of pain. This is a vaginal endometriosis in this picture. I've never seen this one before, but I mean, look at that. That's endometrium. This is the freaking uterine wall growing inside the vaginal canal. Very painful. It can occur almost, oh, this is a great picture. Yeah, barbed wire. Imagine uterine cramps and then imagine it occurring like at its worst every second of every day. That's what I've heard endometriosis feels like. Yeah. You have it right now, Trish? I'm so sorry. God, I just want to hug you right now. Breaks my heart. It brought some tears to my eyes, girl. You take care, okay? Get it taken care of. It doesn't, Heather. Things like that, well, I shouldn't say it doesn't. There's always exceptions to the rule, but that can end up easily causing miscarriage after miscarriage after miscarriage, just because the uterus is going to recognize that it's not ready. Man, Trisha, you can teach me something about it. Yeah. All I can say is big hugs from me to you. You take care. Hopefully you got a good support unit and everything around you too. It's brutal. Quality of life, peeps. It's a big deal. We don't think about certain things on a daily basis, but you know, you don't have to have cancer to have like a horrible life. There's, I mean, don't get me wrong, Trish. I'm sure you have an amazing life, but I'm just, you know, the quality of life and pain. It's one of the reasons why we have an opioid epidemic, right? It has to do with pain. Yeah. All right. Anyways, moving on. The vagina. Okay, the vagina itself. By the way, it's okay to say the word vagina. I get a lot of people that like back in real life class, I'd get so much giggling anytime I'd say penis and vagina. It's just like, you know, a classroom full of 2-year-olds. It was pretty damn funny. I'm scared about clicking on links. Ah, gotcha. Let me get through. It's 12 o'clock. We're not doing great on time, but we're doing okay. Tomorrow we'll be finishing up like ovarian cycle stuff and then sexual differentiation, which is part of another lecture. But what I want to, I guess let's go ahead. There's something called internal female genitalia, and then there's external female genitalia, and of course the same thing goes with males. There's internal male genitalia, and there's external male genitalia, and it's a little bit simpler in males because external is external, whereas in females, internal isn't quite as easy to visualize. Internal female genitalia is basically the upper portion of the vagina. But the lower portion of the vagina is part of the external female genitalia. So everything you guys see here is considered internal female genitalia. So that's why this picture doesn't show the rest of the vagina. Only the portions associated here with the uterus. Maybe this will make more sense in just a minute. The vaginal wall, you guys see the folds? Those are rugae. Rugae, plural, vaginal rugae. Please be specific in your answers. What do we know about rugae? Elastic tissue. The vaginal wall and the vaginal canal are extremely elastic. Thank goodness. There are certain layers to the vagina that we're going to be discussing in just a second. Just like most of the organs of the body, we want to talk about the layers, 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 layers. Good old ogres. Ogres have layers. You don't know that, then shame on you. Instant fail. You mean like a parfait? No, like an onion. The last part of this anatomy right here is the fornix. Now, the fornix, there are lateral fornix, there's an anterior fornix, there's a posterior fornix. I love Shrek. I'll still watch it. I'm just a child. What can I say? 43-year-old child. The fornix. I know I had this in one of my narrations because somebody texted me about it and said that they'll never forget it. This is a booby trap for dumb sperm. That's what I call it. There really isn't any other function of the fornix other than to trap dumb sperm. So if sperm can't find that cervical canal, they're going to accumulate here in the fornix, and the fornix is especially acidic. So there's a lot of extra acid that gets built up here, and it's going to just massacre those dumb sperm. But you can see that the cervix itself actually pushes out into the vaginal canal. And it creates this impression, this space, this area around the cervix, and that's called the fornix. All right? Depending on the position of your uterus, you might have a fornix that's either more shallow or deeper. Just depends on the person. Layers of the vagina. There's a mucosal layer, there's a submucosal layer, and there is a muscular layer, right? The submucosal layer is considered a part of the mucosal layer here. There's basically just, some glands that help with the secretion of mucus, and they're all in this purple band here. Below that, smooth muscle. Okay, great. We need to have some smooth muscle to help with vaginal contractions, both during sexual arousal and during childbirth. I mean, pretty darn important to be able to have these vaginal contractions. And then the outside layer is just connective tissue. It's not in the peritoneum, therefore it's called adventitia. Everybody feel pretty comfortable with this so far? Right? Rugae, therefore elastin. I mean, it's not that complex. Still, this is just something we've already learned. What type of epithelium do we need in place in an organ that's going to have a baby going through it? Do you want a simple single layer of cells with some nice little delicate cilia? Or do we want a phone book? Or do we want a phone book in place? Transitional cells are actually very weak when it comes down to it, Heather. They can stretch, they can get inflamed. Bladders are not, made of steel. No matter what the saying says, that person has a bladder of steel. No, they don't. We need something that's like a phone book, thick and highly protective. Awesome. Now, the external female genitalia, all of this combined is called the vulva. And you're going to be going through all of these different external female genitalia structures for lab, where you're going to be identifying the mons pubis. This is not mom's pubis, so don't write that on your lab practical. This is mons, means mound. This is where pubic hair grows from. You have a labia majora. Labia just means lip. So you have a large or major lip and a minor lip. So labia majora and labia minora down below. Vestibule. Vestibule just means roof. So it's the space, it's the roof, it's the tissue. The roof of this vulva is the vestibule. And you're going to actually be looking at some models that have vestibular glands found inside of them. So inside of here, there are actually a number of different glands that you're going to have to identify. The clitoris. This is the glands of the clitoris that's exposed to the outside. You guys remember me saying something about the glands of the penis earlier and how it's the exact same tissue as the glands of the clitoris. So in a male, because there's some hormones we'll learn about tomorrow that are involved, it's going to end up causing the elongation of the clitoris, and this is going to become the penis in the male. Becomes the tip of the penis in the male. And in the male, instead of having a labia majora, that labia majora fuses and becomes the body of the penis. It's all the same tissue. It's all the same origin. So do you think there is tissue inside of the labia majora? It's actually technically inside of the vestibule and the clitoris that can be engorged with blood, just like the male penis? Absolutely. So keep that logic in place, right? When you're learning about this anatomy, don't dwell on the differences, dwell on the similarities, and it makes it a lot easier to study. The last thing that you guys see here is a triangle. Everybody see this, or a diamond, or whatever the hell shape this is? Triangular. This is I have this as part of my lab objectives, I'm pretty sure. And every semester, students are like, where is this thing? Help me, Dr. H. This is called the perineum. Kind of. Peritoneum, not the same thing. Perineum. So there's a space between the vagina and the anus right there. That's actually portions of the perineum, right? And then this entire triangle right here, This is comprising up the entire vulva with the perineum in the middle of it all. I don't know if that helped. I think I might have just confused you guys. But that's in essence what we're looking at here. From the pubic symphysis, you guys remember the pubic symphysis? It's the cartilage that helps hold the pelvic girdle together. What about the ischial tuberosity? Do you guys remember that one? Probably not. Yeah, exactly, Tariq. The ischial tuberosity, I call my butt bones. They're the ones that you put all the weight on, so they're the big protuberance on the bottom of the ischial portion of the pelvic girdle. There's your coccyx back there. So yeah, that's this triangle, all part of the vulva and the perineum. Okay, cool. Moving on, because we're running out of time. The functions of the female reproductive system are a lot. Female reproductive system is a lot more complex. I don't need to tell you guys this. Everybody knows the female reproductive system is more complex than the males, I think. If you don't, there you go. Female reproductive system, much more complex than males. They do a lot more than just gametogenesis and gamete transport. They have to go through the entire process of fertilization, implantation, nourish and develop a baby, give birth, not to mention monitor and regulate all types of hormones through this entire process. Males, just give me a certain level of testosterone, I'm fine, I'm happy. Not too high, not too little, Goldilocks, good. Females, all over the dang place. So let's focus on gametogenesis for females. And what I want you guys, I should have had that on the, dang it, dang me. What I want you guys to see with the female reproductive system is that it's going to be very similar to spermatogenesis in the male. What do I mean by that? In males, we're making spermatozoa. In females, we're making oocytes. So far, so good? Technically, we're going to end up making ovum in females. We never get to, well, we'll deal with that later. One sec. So in females, we want to make oocytes. In males, we started with spermatogonia. Well, guess what? In females, we're going to start with oogonia. We'll start with one. Just a little bit of practice, and the root of that word shouldn't be too crazy. This oogoneum is going to go through 1,000 different stages of mitosis. It's going to go through mitosis, and then mitosis, and then mitosis, and then mitosis, and then mitosis. Multiple stages of mitosis. And we're going to end up with millions of oogonia. You can already see that that's a little bit different than in the males. Yes, no, maybe so? Because these steps occur before you're ever even born. These steps right here are all occurring during fetal development. When do males start producing new sperm? When will their spermatogonia start to divide? Only post-puberty. That's it. Females start this process way earlier, when they are fetus. Yeah, pregnant women are carrying their grandkids. It's so true, and a little bit really cool, and a little bit creepy. Where are we at here? Right here. So for oogenesis, we start with an oogoneum, which is a diploid, and it's just going to go through multiple stages of mitosis, massive amounts of mitosis, to the point where there are 1.5 million of these cells that will be present when you're born. Yeah, I think that happens a lot. Like we all think we know a lot. Even I think I know a lot, but I don't. I know nothing. I'm Jon Snow. I know nothing. Possible I watch too much TV. So, Oganium. We got before birth, during fetal development, we're going to have lots and lots and lots of mitosis. And those cells are going to end up entering meiosis 1. Well, what did we see in males? It went from spermatogeneum, and it's going to go through different stages of mitosis. And when it went to the first stage of mitosis, or sorry, the first stage of meiosis, what did it become? Oh, that is exciting. What did it become? Good. It's going to become a primary spermatocyte. Why? Because it entered meiosis 1. Yes, no, maybe so? So what's going to happen over here? When an oogonia enters meiosis 1, what's it going to become? A primary oocyte, though. Not just an oocyte. It's entering meiosis 1, so what do we call it? A primary oocyte. Do you see the similarities in terminology? I'm trying to show you the similarities. There's plenty to focus on that's different. So why not focus on what's similar, and it makes it a little bit easier to study. So it's entered meiosis 1. Now, in males, this immediately is going to transition to a secondary spermatocyte. We immediately start meiosis 2. Huge difference between males and females here. In females, what happens right now? Freeze. Okay, these primary oocytes, you have about 1.5 million at birth that are frozen in the first stages of meiosis 1. And then your body's going to go through and start picking these out over the next, anywhere between hopefully 13 years of your life before you enter puberty. Hopefully you make it all the way to a teen, but that's definitely not happening as much anymore. Not a lot of fun being a 8-year-old going into puberty, but it's definitely happening nowadays. And then what's going to happen is that one of these primary oocyte is going to be stimulated, actually a couple dozen will be stimulated. No, yes, they're eggs. Yeah, we'll call them eggs, sure. Eggs are tricky, but yeah. Yes, these are eggs. I know your semantics. I know most female semantics. So yes, these are what most women would refer to as eggs. Got it. And the body's going to start picking these things out. So you start with about 1.5 million of these, and then they're going to start to become less and less as you progress until puberty. But I think I got a slide right here for it. There we go. So at birth, you had about 1.5 to 2 million, and then at puberty, there's about 300,000 left, and you only ovulate for about 400 through your lifetime. So there's a lot of oocytes left. A little bit crazy.
At one point, there's even 6 to 7 million, by the way. I didn't even mention that. By the time you're born, you've gone through about 6 or 7 million, and you've gotten rid about 5 million of them. It's just like, nope, you're not good enough. Nope, you're not good enough. Nope, you're not good enough. Talk about like, the penultimate dating game. No, you're not good enough. Go, go. Nope, don't even like the look of you. Go. Yep, swipe left. God, horrible. Yeah. Okay. So let's see here. Now, what's going to happen at this point? We're frozen in meiosis one. Now we get to wait until puberty. So that's the difference. In males, spermatogeneum don't even start mitosis until puberty. In females, wait until puberty. And what's happening here, some hormone switches are going to occur. And that's going to trigger maybe a dozen, sometimes 2 dozen of these primary oocytes to start to enter and finish meiosis one. It takes a long time to finish meiosis one for a female. Okay, this process takes about 2 weeks. Huh. Sure sounded a lot like the ovarian cycle, huh? So as you start your ovarian cycle, your menstrual cycle, it takes about 2 weeks or so before that primary oocyte has completed meiosis one, at which point it's going to enter meiosis two. And what's this going to be called? A secondary oocyte. This enters meiosis two. But guess what? Females are complicated. Frozen again. This secondary oocyte is what gets ovulated. And it's going to cause those fimbriae to start to sweep back and forth. It's going to create that current, that vacuum that now pulls the oocyte, this non-fertilized ovum, up into the infundibulum, into the ampulla, where it waits for fertilization. When does it get a complete meiosis two? Following fertilization. That's when it gets to complete meiosis two. Hurry up, wait. Hurry up, wait. That's the female reproductive system. Following fertilization, we complete meiosis two. Spermatozoa can now properly fertilize the 23 chromosomes of the oocyte, and we're going to end up with a whole bunch of crazy stuff. But it starts out as a zygote. Zygote's going to go through a bunch of differentiation phases, and it's going to end up becoming a blastocyst and a morula, and gets crazy. Yep, hurry up and wait. Do you guys see the similarities between the male and the female system? And do you guys see the differences? Of course, there are a lot. I just don't want you guys to focus as much on the differences because it can make this way too complex sometimes. Focus on the similarities, then work on the differences, okay? Okay, female reproductive system. much more complex. You will have to be looking under a microscope and identifying different phases of what's called follicles. So what is a follicle? All we talked about up here is a primary oocyte. Down here, we talked about a secondary oocyte. We get this ovulated oocyte, right? What do we say? The primary oocyte's going to develop, and it's going to develop, and it's going to develop, and it's going to develop. When the first stage of meiosis is done, boom, it's a secondary oocyte. But now we're frozen again. And we go through ovulation, and that secondary oocyte isn't completed until that spermatozoa fertilizes that mature ovum, at which point now all of these different chromosomes can make that baby. But what really is going on here, it's not just an oocyte, it's not just a primary oocyte, it's not just a secondary oocyte. If we're going to have all these different stages of getting frozen in some stasis, I hope you guys can see that this cell needs to have protection. Yes, no, maybe so? Yes, and exactly. Follicle is a cavity. It's a container. It's a storage area, and that's what is going on inside that female reproductive system. Is we have follicles called ovarian follicles. They're going to be made up of the oocyte, primary or secondary, doesn't really matter, and they're going to be surrounded by follicle cells. Follicle cells are going to turn into a number of different types of cells. We're going to discuss just a second. And these follicle cells are found inside of the ovarian cortex. That's where, when you're born, there's 300,000, well, not when you're born, sorry, when you enter puberty, there's about 300,000 what's called primordial follicles found inside the cortex of the ovaries. Primordial. A primordial follicle. Primordial tells us that it's, of primordial origin. It's the most primitive type of ovarian follicle. This is being created during fetal development. So we go back here and we think about what else is happening during fetal development. We have mitosis, and it's going to be arresting these primary oocytes in the very first step of meiosis 1. And then these primary oocytes get packaged inside of cells. As they're frozen, they get packaged inside of cells. These are primordial follicles. Did that make sense? So the whole structure is a primordial follicle. The primordial follicle is going to be comprised of follicle cells that surround a primary oocyte. Perfect. Perfect, perfect, perfect. Love it. Yeah, I mean, I literally was awesome. Segue. I was on my way to go to Slidebox. No, oops. It's all right. It was good timing for it. That's why I thought of it at the same time too. I was like, this would be a good time to show them these, so we don't have to do it again later. So the female ovary, kind of funky looking, really, when you take a look at a slideshow of this, right? There's a medulla and there's a cortex. Shocker. At this point, that shouldn't be too off the wall, right? The cortex is the outer layer. So if we start looking at the cortex, what are you doing going so far? We start looking at the cortex. You can see there's big circles and there's little circles. You guys see these little circles? Zoom in. Zoom in. This outer edge is the ovarian capsule, just like the renal capsule. Right, this is an outer connective tissue that helps hold everything together. Right along the border of that, we have some very, very, very small structures. If I keep zooming in, you'll notice there's a big purple dot in the middle that's surrounded by flat purple dots. This is a primary oocyte surrounded by follicle cells. Yes, no, maybe so? You guys see it? It's a primary follicle surrounded by follicle cells. a primary oocyte surrounded by follicle cells. That's where we find primordial follicles, right there, all along the border. So let me zoom back out again. And this is just one tiny little slice. And can you see that this outer cortex is packed full of primordial follicles? Yes, no, maybe so? Just absolutely packed. Cool. Next, what's going to happen is the primordial follicles are going to get stimulated by a hormone. What hormone could possibly stimulate a follicle? That just doesn't make any sense to me. Oh, unless the name tells you, right? This is follicular stimulating hormone. Everybody see the word, understand what it's actually doing, how we identified this hormone. It's a hormone that stimulated follicular development. So now these primordial follicles are going to start to develop, and they're going to have, a dozen or maybe 24 of these primordial follicles that are stimulated during each cycle by FSH. And as that FSH is in the system, it's going to cause the primordial to become a primary follicle. Primary follicle. Make sure that your brains are differentiating follicle from oocyte. You'll see why in just a second. The primary follicle, what we start to see is that the primary oocyte starts to enlarge. That's one of the first things you start to notice in a primary follicle versus a primordial follicle, is that the primary follicle starts to enlarge. What else happens is the follicle cells surrounding the primary oocyte, they start to differentiate, and they become two different types of cells. So the follicle cells become one, what's called follicular cells. Nice. But we're not going to call them follicular cells. We're going to call them granulosa cells. Not granular cells, granulosa cells. You can see that bold right here. So there's a whole bunch of cuboidal follicular cells that start to emerge. These are called granulosa cells. We'll come back to what they do in just a second. Those follicle cells also become what's called thecal cells. Be sure to pronounce that very well. Articulate it. I'll never forget the first time I didn't articulate it very well, and I was like, thecal cells. And somebody was like, did you just say fecal cells? No, these aren't poop cells. We're good. They're thecal cells. Thecal. So let's get to a picture real quick so this makes a little bit more sense. The peripheral follicle cell, you can see, are these kind of flattened. They're cuboidal, but they're a little bit more flat. These are thecal cells. And underneath that, we get multiple layers of these granulosa cells, a lot more cuboidal. Yes, no, maybe so? And the way this works, actually, let's do, well, we'll grab one of these arrows. The way this works is that thecal cells secrete androgens. Do you guys remember that androgen is testosterone? Tried to emphasize that earlier today. And we know testosterone and other androgens are nonpolar. So as these produce androgens, these androgens just diffuse into the granulosa cells underneath it. And then the granulosa cells take and convert the androgen into estrogen. So there's a flow chart. Thecal cells produce androgens, and then those androgens are converted into estrogen by the granulosa cells. You guys have that written down? I don't understand what you mean. Thought androgen can be either or. Androgen is androgen. Androgen is just a chemical. And then androgen gets converted. Androgen is androgen. No, androgen is not estrogen. So if you hear the word androgen, you should not be thinking of estrogen. However, if you hear the word androgen, the body, females especially, are converting androgen into estrogen. So the more androgens you have, exactly, it's a precursor. You got it. Yes, you got it. I'm sorry, I didn't quite understand what you meant initially, but that's 100% true. It's a precursor hormone. All right. Good. Yeah, absolutely. You guys both got that there. Yep. Yep. Lots of different therapies that we use using androgens as well. That's male, of course, because androgens are important for both males and females. All right. We're losing track of what's important here, though, and I'm running out of time. So thecal cells produce androgens. Androgens get secreted into the granulosa cells, and the granulosa cells convert those androgens into estrogen. Yes, no, maybe so? And it's the estrogen in combination with FSH that is going to allow this follicle to continue to develop. This primary oocyte is getting bigger. It's enlarging. Okay, it's actually it's swelling up. It's getting bigger as it goes through meiosis 1. The cell is getting bigger and bigger and bigger, and the number of layers of granulosa cells is going to increase and increase and increase. And if there's more granulosa cells, what's going to happen to the production of estrogen? Increase, decrease, no change. So now let's start looking through this. There's a bunch of crazy going on here. But what you'll notice is near the edge, we have primordial cells over here. But right here and right here, what I have is a enlarged primary oocyte with a layer of cells in between and some thecal cells on the outside. Yes, no, maybe so? Even here, this is starting to go through the process. This one right here, all of these that I just pointed out, those are primary follicles. So the primordial follicles are here. And these ones out here are primary. Are those small purple ones here? You mean these? What do you, no, those are going to be thecal cells. That's the outer border. The granulosa cells don't have quite as well-defined nuclei. If you actually look here, now we start to get some more defined nuclei on those. But yeah, at that stage, just not going to be as quite as defined. All right. Let me double check that I'm not completely lying to you. Actually, I don't mind saying that I think I was wrong right there, because you can see this dark purple band that surrounds it. Those are going to be the thecal cells. Great catch there, Heather. I knew this. I just, yeah, it is what it is. So, and then these ones right here, granulosa cells. Great. Yep. And then the primary follicle right in the middle. Cool. Awesome. Now, next goes from, and we've done this, right? We did this. We got a primary oocyte, and it's going through these different stages of meiosis 1, but there's a lot that goes on in between. So now we can start to add a flow chart in here, right? Is that until puberty, well, now we're going to have a primordial follicle. Let's do this. Over here, we'll have a separate flow chart for follicular development. And it's going to start with a primordial follicle. And this is going to become a primary follicle. It's not the hardest. Should probably, hopefully, be one of the easiest when it comes down to studying. Primary follicle is different because thecal cells, granulosa cells. So what's going to happen next? Now it's going to become a secondary follicle. I need you guys to be paying attention to the terminology. A secondary follicle, not a secondary oocyte. This gets confusing for students. A secondary follicle still has a primary oocyte. A secondary follicle still has a primary oocyte. Is that clear? Does everybody have that? Make sure you check yourself before you wreck yourself. This is just a continuation of the follicular development. The primary oocyte's getting even bigger. Look what's happened to the number of granulosa cells. There's even more of them. So what's happening to the amount of estrogen being produced? Increase, right? You still got your thecal cells surrounding this. One of the defining parts of a secondary follicle is the presence of this fluid-filled space. This is called an antrum. Okay, so as the follicle has been developing, it's producing tons of estrogen and some other fluids and stuff, and it's going to start to get to the point where we have a fluid-filled chamber, an antrum. Antrum, A-N-T-R-U-M, antrum. What's in it? Fluid, including a lot of stored estrogen. Lots of stored estrogen and lots of secreted estrogen as well. Okay, so there's fluid, it's a fluid-filled antrum. This is a pretty defining portion of a secondary follicle. So if we go back to our stuff here, you don't see an antrum here. You don't see an antrum here. You don't see an antrum here. What are you starting to see in some of these other follicles? What do you see? Help me help you. See an antrum. Now, there is no antrum here. So is this a secondary follicle? Nope. What you're going to notice is the antrum gets bigger and bigger and bigger as it develops. This is a great example of a secondary follicle right here. You can see that there is an antrum. It's not too big. The follicle itself is still relatively small. On a lab practical, I would show a picture that has a lot of different follicles in that picture sometimes. Other times, I'm just going to ask you guys or expect you guys to be able to look at the size of the antrum and make a judgment call. So over here, would this be a primary or secondary follicle? Kind of. So this is a secondary, exactly. So what's going to happen next is that this antrum gets huge. All right? We're going to come back to what's going on here with this cumulus O force in just a second. But what's going to happen next is that that antrum gets really, really, really big. This is called a mature follicle, at which point the primary oocyte finishes meiosis I. So when we get a mature follicle and we finish meiosis I, what is the name of the oocyte is going to become? If we finish meiosis I and we start meiosis II, now we have a secondary oocyte. Does that make sense? So there's no secondary oocyte until we make it to a mature, also known as a graphian, G-R-A-F-F-I-A-N. Yes, no, maybe so? All right, cool. Now, we take a step back, I need to show you guys this cumulus oophorus. So this whole structure you see here, this peninsula of cells that surrounds the oocyte. Well, these are granulosa cells. Sweet. So we have these cells now that are surrounding the oocyte, kind of pushing out into the antrum that still produce estrogen. But there's something else going on. Oops, I was trying to zoom in and I just zoomed past. Is that you'll notice that there's a space. The primary oocyte is defined by the purple line. The granulosa cells are out here. So what's right here in the middle? This is called the zona pellucida. You guys remember the septum pellucidum that separates the right and left ventricles in the brain? It's the same thing. Pellucida is just a soft tissue proteoglycan layer. So proteoglycans, these are proteins and they're carbohydrates. They create both an energy source and a protective barrier. So the primary oocyte is starting to get protected. Don't overthink it. You will need to identify it, zona pellucida, versus the granulosa cells, which are called the corona radiata, the radiating crown. Okay, so it's a crown that radiates all the way around that primary oocyte. So now let's go over here, and you can start to see these structures pretty darn well, I think. Right, so here we go. We have a secondary follicle. There's the antrum. This whole structure right here is the cumulus oophorus. And if I were just to point at the purple layer out here, that would be the corona radiata. Does that make sense? And yes, it means clear. Does this make sense? That is the corona, the purple cells out here are corona radiata. And then where my mouse pointer is now, that's the zona pellucida. And then where my pointer is now, it's encircling the primary oocyte. Yes, no, maybe so? Awesome. Now, technically, only one follicle makes it to mature every single menstrual cycle. Kind of crazy to think about it. You get all of these follicles all pretty much ready to go, and then the body's going to say, nope, not good enough, not good enough, not good enough, not good enough, not good enough. Only one of you is good enough. And that's between both ovaries. It's a hard knock life. So now we got this right here. And then with a big old endocrine surge, this secondary oocyte is going to be ovulated. And what gets ovulated is not just the secondary oocyte, it's the entire cumulus oophorus. That's why it's an important structure to understand. The corona radiata, the zona pellucida, and that secondary oocyte, because the corona radiata keeps providing this oocyte with estrogen, and the zona pellucida helps to protect it and give it some energy. The whole thing is called the cumulus oophorus. Words right there. Cumulus oophorus. Boom. Now we're going to go to ovulation of that entire structure. We're not done. Of course not, right? Why make it easy? What we're left with is a huge, open, hemorrhaged, ruptured space. Imagine, again, the nastiest zit you've ever had. Don't tell me you've never had a zit. That's not true. And now you get this just like postulated opening, basically, where that zit just erupted. Well, your body, yeah, that's such a weird show. But your body now wants to use this. This is usable material. We got granulosa cells we've been working a long time to be able to produce. Let's use it. So we take this. This is called a ruptured follicle. And we take that ruptured follicle and we start to fill it in with triglycerides and some other nutrients and stuff necessary to run this system. And as we add triglycerides, it becomes yellow. And in yellow in Latin is luteum. Luteum. Corpus means body, so this is a yellow body. Corpus luteum. So after the follicle has ruptured, once we have ovulated, the ruptured follicle gets filled in and it becomes a corpus luteum or luteum. And this corpus luteum I always test on because we're not done using this crazy thing. We need to communicate with the uterus and with the brain. Hey, I just did my job. Time for you guys to do your job. The corpus luteum keeps on secreting hormones, buckets of hormones afterwards. This is where almost all of the progesterone in the female body comes from. Okay, almost all progesterone is produced from the corpus luteum. Corpus luteum continues to produce estrogen. We're going to go through a graph tomorrow. You're going to see it's still producing a lot of estrogen. So as that ovulated egg is sitting there in the ampulla of the vas deferens waiting to be fertilized, progesterone levels are going to be high. Estrogen levels are going to be high. And in addition, and you can see bold capital letters down below, look what else is just secreted by the bucket load. Inhibin. And what does inhibin inhibit? It stops FSH, not estrogen, Heather. Inhibin only inhibits FSH. Do we want more follicles to be stimulated if we already have an oocyte waiting for fertilization? Hell nah. We want to stop all follicular stimulation. We want to stop the process of follicular stimulation, follicular development, while we are trying to focus our energy on that egg and on the uterus. Does that make sense? So what happens if the egg doesn't get fertilized? The way I like to put it, the corpus luteum throws a temper tantrum, and it becomes a corpus albicans. It's angry. It's angry. Albicans, albino, white. We got a white body. As those triglycerides get used up, and as that corpus luteum is no longer required, we end up taking, regressing, and eating this structure, and it becomes this white body that's left over. It's called the corpus albicans. No longer secreting progesterone, estrogen, and inhibin. So what's going to happen to the inhibition of FSH? We're going to go through a big flowchart on this tomorrow. If we're not producing inhibin, we're no longer inhibiting FSH. So FSH levels start to go back up, and we start stimulating follicles again. Does that make sense? That's just one small part, but I think you guys can follow that pretty easily right now. Great flowchart that follows all of that. So when you guys go to study, now this should make a little bit more sense. Before birth, we start with an oogoneum that goes through multiple stages of mitosis. We end up with millions, 6 to 7 million of these primary oocytes arrested in meiosis 1, the first portion of PMAT is prophase, so it's getting frozen right there. And packaged in follicle cells, so we end up with a primordial follicle. When you're born, there's about 1.5 million of these things left over. And by the time you reach adolescence, maybe about 300,000 left. Still plenty, because hopefully nobody here has 300,000 babies. Once it's stimulated for development by FSH, the primordial follicle becomes a primary follicle, thecal cells, granulosa cells producing estrogen. We develop. That primary oocyte is enlarging, it's getting bigger. the antrum forms in a secondary follicle, and that antrum's huge in that mature follicle. That primary oocyte finally becomes a secondary oocyte, at which point it's frozen again. And we wait for some hormones to cause ovulation. We pop that zit, we start to add some fatty tissue to it and some other things, but it ends up becoming the corpus luteum, which is producing progesterone, estrogen, inhibin, communicating back with the uterus and the brain, hey, do your job. It's going to finally regress and become a corpus albicans. Just because this is a tough one for students to see, is I want to make sure people realize that the corpus luteum takes over the entire ovary, basically. So when we're in the last portion of the ovarian cycle, when we've shut down follicular development, look at what's happened. This is that same ovary, but now instead of having all of these follicles, there's still kind of this big open follicle over here, but most of those follicles are gone, right? This looks like gigantic, just mush. It looks like hamburger. It looks like a brain. Yes, no, maybe so? That's because it's been taken over by the corpus luteum. Look at this entire border that I'm circling right here. Everybody see this? All of that. That is all the corpus luteum. All of that is the corpus luteum. What are these little pieces of white circles over here? What are these going to be? I know they're pink, but relative to the purple corpus luteum, these are white. What are these? Help me out. All of these are corpus albicans. Does that make sense? So this would be how I would test you guys on corpus luteum, and I always like to do it. So I'm going to give you a, it's going to show almost the entire ovary, and it's just going to look like one gigantic hemorrhage, because that's what it is. Great question. The albicans is still going to form, but that's only once the uterus takes over. So once fertilization happens, we still need to get that fertilized egg out to the uterus where it's going to implant in the uterus. And that is not an immediate process. That takes days. It takes upward of 8 days to get that. I got actually a pretty good picture of it somewhere around here, right? It takes upwards of 8 days to get that fertilized egg as we start to divide it, goes through all these cellular processes before finally we get this thing called the blastocyst that is going to implant itself into the uterus, and then a short period later, it's called an embryo finally, where you get the little alien growing inside of there. Yes, alien, deal with it. They're the cutest little aliens ever, though. So there's still a long time for that corpus luteum to have to work. Does that make sense? We still need lots of progesterone. That progesterone is talking to the uterus this whole time. It's causing that uterine functional layer. Remember me talking about the functional layer? This functional layer is growing because of the presence of progesterone and estrogen, but primarily progesterone. So this is where that blastocyst is going to implant, is right into that functional layer. Okay, you guys have a lot to study. A heck of a lot to study. Sorry. If it helps, I love y'all. I'm not sure that helps much, though. Don't forget, you guys have this drag and drop. Most of you should have already done the same time frame. I'm not sure what you mean. Not weekend. No, Unit 4 will be due Friday night. Right? Isn't that what I had? Let me double check that. No, Unit 4 homework is Wednesday. Unit 4 is Thursday. It's Thursday, y'all. Yep, it's Thursday. That's why I have this. SG due for repro. Yes. You should have already done that. Yeah, I got you. I'll still accept study guides for repro if you guys haven't done them yet. That's fine. Yeah, definitely for extra credit. So I'll open it back up again if you guys didn't submit it. I'll open it back up again. Oh, it might still be open. Nope, not open. I'll open that back up after class. Oh, no problem. I just want you guys to study. I don't really care if you guys get extra credit outside of the time frame. It don't matter. Do the work. Get the grade. All right. Comp is Sunday. Well, anytime between Thursday and Sunday. You can take it anytime. I have to have grades posted on Monday. And I want to make sure you guys have as much time between your Unit 4, whatever you need to study. I just want to give you guys as much of a breather as possible during a crazy summer course. Do you guys remember when you were signing up for this class that there are 5-week AMP2 classes? Can you imagine learning everything we did in 8 weeks, but in 5 weeks? It exists. 5-week AMP2 courses exist, and there's no taking your foot off the brake. Yeah, exactly. Like I think in 2 months you can actually retain a bunch of what I'm teaching you. I think in 5 weeks it's really hard. Like you can learn it, you could purge it, you could put it on a test, but are you really gonna hold on to that for the long term? I don't know. Let's see here. Don't forget there's a drag and drop, right? So some of this stuff right here we covered in today's lecture. Is anybody completely freaking out by what I'm doing here? Actually, that's wrong. You guys recognizing these terms? Fundus of the uterus, body of the uterus, lumen of the uterus, shouldn't be too hard. Cervix of the uterus. We start looking at things like this, side views. Please take your time to go through this because being able to look at the uterus from a posterior or an anterior view is definitely important. Looking at things like the mons pubis, pubic symphysis, vaginal orifice, just because we saw them with one particular view in lecture doesn't mean that that's the only way that I will test you. So make sure you're looking at all of these different views, all of these different angles. In terms of labia, students tend to get these wrong because they think that labia majora should be this big, huge labia here, but in a cross section, it's not. Okay, the larger of the labia is on the outside, but you can't see it from this mid-sagittal view. Yeah, the mons pubis is this yellow, and that's because that's where a bunch of triglycerides are stored right there, right? So yeah, definitely a place where triglycerides can decrease their size pretty easily. So labia minora here. So these are just a couple ones that students do have trouble with. Here we're talking about cervical canals, internal osses and external osses, and some fornixes going on here, fornices. But I find that students get the labias wrong a lot every semester. So I wanted to point that out to you. I think most of the rest of these we covered, right? Everything from urogenital diaphragm, doesn't matter whether it's male or female, it's just this muscle layer here. Different layers, endometrium, myometrium, perimetrium, rugae, vagina, vaginal orifice. Do you guys all feel like these are words that you've heard before and that you just need to practice? Isthmus, fimbriae, infundibulum. Okay, so I'm going to go ahead and let you guys do what you do. I recommend when you're going through these personally, I like flowcharts. So like I would start down here in the testes and I'd go, well, down here in the testes is where I know that we have seminiferous tubules. Now, there's no seminiferous tubules in here. And then those are going to go into the red testis, and from there, they're going to go into the epididymis. So that's how I like to label when I study, from the epididymis into the vas deferens. This kind of keeps me organized, so I'm not just randomly putting answers down all over the place. Practice? We talking about practice? Which practice are you talking about, Andy? That one practice? You're just talking about the reproductive drag and drop? Oh, okay. I'm sorry. Practice? Yeah, so and then here's a model of the ovary. So you can see everything from the corpus luteum, corpus albicans, the primordial follicles, primary follicles, secondary follicles, a mature follicle, a ruptured follicle. Not too crazy, right? All of these we just walked through, white, yellow, different sizes. Am I lagging? I'm trying to get through this for y'all. You could not differentiate? Okay, good. Thank you for pointing something out. So if I do something like this, that's the spermatic cord. Vas deferens is just a tube. So it's inside the spermatic cord, like right there. So that pink right there, that's the vas deferens. Whereas the cord is the whole dang thing. Whereas if I'm pointing to a space right here, now we're talking about the inguinal canal. Make sense? There's also portions of that vas deferens that we can see even, actually, no, that's not it. That's the ureter back there. Ureter hanging down from the kidneys and attaching to that urinary bladder. So yeah, that's what we're looking at right there. Okay, cremaster muscles, they are found on that spermatic cord. The dartos are found inside of that scrotum. Yeah. Oh, you know what I didn't do is monkey face. All right, we got to do monkey face. Monkey face? What you guys talking about? Dr. H, you cray. I'm talking about the monkey face. Testes. Man, they don't do a good job here. Where's my monkey face? Seminal vesicle, seminal, prostate. Oh, penis. Here we go. So you guys are going to have to look at histo of the penis. Look, we got a monkey. Ooh, ooh, ah, ah. We do a cross section of the penis. You got these big old monkey eyes, and you got a monkey mouth. Easy peasy lemon squeezy. There's that monkey mouth. That's the urethra right there. There is erectile tissue around the mouth. There's erectile tissue around the eyes. 1, 2, corpora cavernosum. Those are the eyeballs. And the spongy urethra goes through the corpus spongiosum. Corpus is singular, corpora is plural. So 1, 2, corpora cavernosum, and 1, corpus spongiosum. Yeah, monkey face. You ever seen Kanye West with those really crazy big sunglasses? Kind of looks like those sunglasses that Kanye West wears too. Student pointed that out to me like 5 years ago. I LOL'd. All right, cool. Yeah, man, he's still mega rich. It's all good. Okay, everybody, it's 1:12. I'm gonna go do an interview for getting pre-check for flying I hope they don't ask me too many complicated questions. Like my name. I'll get this thing unlocked for you. If there's anything else you guys need from me, let me know. And I will see you tomorrow. Tomorrow, we're going to be going through the rest of the female reproductive system, how the endocrine system works, and sexual differentiation. So be prepared. Be prepared. Adios, y'all.